Hey everybody, good morning from RV Falcor 2. You're watching the dive stream, and this morning we're at a new site called San Antonio Canyon. So we're really excited about this dive today. We're gonna see what we can find. Right now it's just a little bit after breakfast. I'm hearing myself, hold on. Speech jammer. Okay. Thank you. And with me here in the control room this morning, I've got a whole array of crew, technicians, and scientists. So right now, piloting, we've got Tyler and Alex. I've also got the one and only Juliana sitting behind me. And with me here in the watch lead position today, I've got our wonderful mapping expert, Adam, and he's going to introduce himself real quick and get the show started. All right, over to you.
All right. Good morning, everyone, and welcome aboard the Falcor 2, and welcome to San Antonio Canyon. I'm Adam Skarke. I am a marine geologist from Mississippi State University, and I'm on board the Falcor to help out with mapping the sea floor. And we've been able to use these maps we're creating to pick some great dive sites like the one we have today. We are in the base or the bottom of a deep canyon here that is cutting across the Chilean margin, and we're going to explore a little bit on the floor of the canyon this morning, and then after perhaps a half hour, an hour, we're going to start to proceed up the wall of the canyon where we expect to potentially find things like exposed rocks and unique ecosystems on those steep canyon walls. So it should be an exciting dive today, and we're really glad to have everyone joining us and looking forward to sharing it with you. So one of the things we've seen quite a lot of so far today on the bottom has been these swimming sea cucumbers, these holothurians. They are, I'm not sure I've ever seen them quite this abundant. They're everywhere down here, which is great. I know one of our science PIs on the vessel, Greg, was very much looking forward to finding some of these, so he'll be excited to see this. As we make our way along the canyon here, you can see it. The seafloor, of course, is quite sedimented, and the water is a bit turbid as well. We know that canyons like this serve as pathways for sediment moving from the shoreline and the continent out into the deep sea. They're a concentrated path for that material to move out into the ocean, and so we may be seeing the effect of that a little bit today in the cloudier water here at the base. But likely that means as we move up the walls of the canyon later today, we'll see that water become much clearer as we move away from the base of the canyon.
Are we returning back to the line and then going up to waypoint 2?
Yeah, we can turn around here and go back to waypoint 1.
Okay.
That works.
That works. Yeah, we're about 70 meters from waypoint 1. Yeah, we're only about 60 meters from the waypoint. Do you want to do a full 100?
Oh, yeah. I'm sorry. I thought we were. We can keep moving in this direction. That'd be
Before we
great.
get to the waypoint, because we're just strategizing what to do with the boat. How to move the vessel, so are we gonna come back to waypoint 1 or go to waypoint 2 from?
Oh, I understand what you're saying. I suspect we'll come back around to waypoint 1. So we'll probably maybe go down and move out into the canyon a bit and back towards 1.
Okay.
Yep.
Just wanna try-
Thanks for Claire. Gotcha.
Yeah, if it's too much, that's the plan. Yeah. Took out 1,100 meters of fill lines. That could be good repair orders. At this point, probably 60. Oh, really, it's like, I know, right? Yeah. Remember that reference? Yeah.
So we're currently making our way along the axis of the canyon just to characterize what the sedimented base of the canyon looks like. We're not quite in the flattest part of the canyon here, but we're down towards the base and getting a sense of the organisms that are here, what the ecosystem looks like before we transect upwards. And I think we're going to explore and characterize this bottom part of the canyon for a little bit, and likely we'll be taking some push core samples a little bit later to get samples of the material here on the seafloor. A little hard to see what we have there. Marcella, do you know what those are?
Corals and a sponge.
Corals in a sponge?
Coral and anemone.
Anemone. Very good. Starting to see some denser aggregations of these corals and anemones here. And it's worth noting we've been primarily focusing on seep environments for our first sets of dives here on this cruise of the Falcor 2, and this is a little bit of a change in our seafloor environment and ecosystem that we'll be observing on the dive today. Oh, it looks like a crinoid maybe that we just went by. That's unique, stocked crinoid. The objective of today's dive is to characterize this deep sea canyon. Canyons are unique deep sea environments. They oftentimes provide locations of steeper walls and in many cases exposed rock, which is important for a number of organisms that attach to the seafloor to have a hard substrate to attach onto, particularly these sessile organisms that attach to the seafloor and don't move. And so canyons can provide that where flat parts of the deep sea cannot because they're covered in sediment. And so we tend to find unique environments in canyons. Additionally, those steep canyon walls cover significant vertical change in depth in the ocean, which causes zonation in the types of organisms we find there based on differences in temperature and salinity. Canyons can also be locations of upwelling or downwelling that can potentially bring in nutrients to these environments, driving ecosystems as well. So a lot of good reasons to explore canyons as we're seeing some corals here. What looks to be maybe an octocoral, maybe a paragorgia, and some crabs as well. Wow, that's pretty cool.
It's possible to collect a piece of coral. This,
Piece of coral?
yes,
Mm-hmm.
maybe it's a plexauride or a candogorgia.
Aconchagorgia? Yeah, I've heard of that one before. Fantastic. It's good context.
Yeah. I want some lowers. Oh.
It's a parking brake?
Yeah, put the anchor down.
Cork would drive
No.
How big of a?
The red one with yellow. The red
What size?
maybe two branch
So by what size?
or coral clip.
Two branches.
All right, so we're getting ready to sample this coral. It's exciting. I haven't been here for a sampling event yet.
On the side one. Ah. Coral is okay. And it's for you? Yes. Le ponemos en la mesa, ¿no?
Very nice.
Just a version. What's your just version? What's your usually version? Like
Oh, are you trying to make me
Oh, me and
I was-
Tyler
I
are
was
fighting.
fighting.
Sorry. Just gonna log yet another It's me.
happy?
Yes, thank you.
See what I'm doing. Or just turn off the photos. Iris will do it.
Just crank myself, just so you can see. Otherwise it would be more difficult.
Hmm.
Yeah, it's super good. Depth perspective is very interesting.
I mean, I can
Just
also.
float
I
off
can
bottom.
Yeah, that's what I was just gonna say.
Yeah, you could raise off bottom and
Do
back.
that.
It's like driving in a snowstorm out here.
Yeah. Alright, coming up. The ship was out of this.
I'm sorry.
Yeah, let's do it. Should be-
14 seconds.
Yeah, we should start out. This is almost turning, so
Okay.
I'm gonna wait till
Yeah.
8 second call right now.
Breaks the sample list if we have hundreds of samples, so
Alright, let me get that somewhere else.
minimize it. But do we need to?
That's all I needed to know.
Yeah, like it's stuff that-
Just.
Carl's getting a ride of his life. Never had this experience before.
So you put pot on the coral.
Yeah.
It's just hanging out having breakfast and all of a sudden
Grab.
pulled up into it.
I want station keep, but I can float. I'm gonna push it out like you. I know I'm saying I can push it out before it's a crash. Yes.
So we're just basically a launch site. We're at the launch site and we came out to the west riders transected. And I saw this coral. It's transector. Was it in the transector? 100 meters.
Yeah, we did.
And it was as dark as a confirmed, but it'll be video. Well, you better, you have to go back. We're planning to return to the waypoint 1 before going out. You may want further out up to the. Because I can't Good morning. We're changing the watch now, the science watch. I'm Lisa Levin from Schmidt Institution of Oceanography. And we have, well, we'll wait till everybody's here. We're in San Antonio Canyon at a depth of 1,065 meters. Looks like we're waiting for some better visibility. It's a pretty turbid canyon, apparently. This is a canyon and Seeps expedition, and up to this point, we've been working on Seeps at Limari, where we didn't find any, and El Quisco, where we found many Seeps, and now we're into the canyon phase. And San Antonio is the first canyon. My understanding is this one is being explored, and for comparison to canyons to further to the south, which will be a large focus of the next leg of this expedition led by Patricia Esken. That previous lighting kind of makes it look like the Milky Way and outer galaxies. The stars. Probably as many stars as there are suspended particles in the water column. Although I'm not an astronomer, so don't quote me on that one. So the canyons are narrow channels that are cut by past rivers, pretty much when this area was on land. Although I believe they can be cut in underwater, but mostly they form on land, and because they're narrow, they focus the water and conduct it, so you can have stronger currents in channels, in canyons, than you can on the non-canyon continental slope. And when you get stronger currents, sometimes you get suspended sediments, especially in a tidally. And you also get a lot of filter feeders, animals that rely on currents for moving food particles and moving the food prey by them. Steep walls of canyons tend to have large gardens of beautiful filter feeders. Okay, we're back down to the axis of the canyon. Could you do me a favor and when we're stable, just zoom in on the sediment itself so we could take a look at what's in there, talk a little bit about that. So the sediment looks featureless, but of course it's not. There's all sorts of things going on down there. Lots and lots of animals probably live subsurface. They would be polychaete worms, small crustaceans like amphipods and isopods, perhaps some mollusks or small echinoderms. I know
we're trying to just zoom in and take a look at the sediment surface. I know I saw some fecal casts, which are what deposit feeding worms deposit out their back end.
Is that just for the vaccine? Yeah. Just the tail. Manually, so I'm just pushing back here. But there we go. That's good. Yeah, I think it's I ran that. Let's do that. And up there.
We're zooming in right now on what looks like somebody's fecal cast. It could be a tube, but I don't think it is. You can see these coiled spaghetti-like structures. Those would be basically the feces of deposit feeders, and I'm not quite sure which worm would make these. Could be a sea cucumber. If we look down, there's lots and lots of piles of these. And that they just tell us that there's a lot of activity going on below the surface that we don't see. At some point, we may see a surface manifestation of these animals, but I don't, which is the fecal cast, but I don't see the animal itself. There are subsurface burrowing sea cucumbers like Mulpidea that can make these. There might be some polychaete worms, possibly. But this is I just wanted us to have a look at what makes that sediment surface look a little gravelly or uneven. It's not actually pebbles or anything. It's little piles of deposit feeder fecal casts. And hopefully, when we take push cores, we'll get the animals that made these. Guys, got a picture. Loggers? Yeah. Okay. Let's introduce, why don't we have our two loggers introduce themselves? They're here.
Good morning, everyone. I am Paola. I am this watch data logger along with Erin, who will be introducing herself very shortly. I am from a student, a guider student from University of California, Santa Barbara. And I am here also representing Dr. Gerber's lab. And we are mainly interested in collecting sediment samples to study microbes within the mud, which get really interesting when we're around seepage area and more so canyons. Erin?
Hi, I'm Erin Fruits. I'm with the Marlowe Lab at Boston University, and I'm on board as a graduate student studying deep sea geomicrobiology. So I'm interested in what microbes are at salt marshes and what they're doing. So to get at that, we're looking at the geochemistry of the seep areas and the diversity of microbes in the sediments. And then we have ways of investigating which microbes are doing what with which chemicals.
Introduce yourselves.
Maybe.
And Sophia.
Carol usually introduces. You need me to speak for you? Yeah.
All right. If you, hold on. Think about how to do this.
We're parts that we can move, actually.
If you
It's
can,
easier.
Lisa, if you can tilt your mic more towards Tyler, yeah, and just project a little bit more, and we should be able to hear you, Tyler.
Nice.
All right. I'll use my teacher voice.
Go for it.
I'm Tyler Smith. I'm currently flying the sub. I'm from America. And this has been a fun cruise so far.
Yeah.
Basic. Project, Alex. You can do it. You're closer. All right, my very shy co-pilot is Mr. Alex Paris. California. He apparently doesn't like to talk, but he's going to be running the manips, and if we do some push cores, he's going to have some fun with that as well.
Great, thank you. Okay. Well, this does look like an interesting area to push core, but we are going to wait for our chief scientist, Yulo Soto, to give us the thumbs up on push coring, because these are going to be his. Hey, look who walked in the room. It's Yulo. You like this spot? Lots of casts, eco casts and everything. Okay, it's push core time. Let's have a push core party.
Excited? You don't need to see you.
So we
You actually just can't see.
we have a request for 11 push cores, all those together. So we'd like to put them in before we take any out.
You know what, since this
And-
is just starts here, do you want me to
Yes.
find a clean spot?
Yes, a clean spot would be great.
So.
We're at 1,077. We're on the flat part of the bottom. Or are we up
It's-
against the slope right now?
So the Matthew says we're on the flat, but it's pretty steep. So I can
Okay.
back off to
I
the very
think
bottom.
what we want to do is be on the axis, on the flattest part of the bottom, so that you don't have to core on a slope.
If you're flat, then you can just land on the skids too.
Is that okay? Apparently, we're on the slope Yulo here, so I'm gonna have them go back to the flatter
Okay.
spot. Although maybe all those burrowers like the slope, I don't know.
You can bounce back up if you want to. If the
Let's
flat's
see,
okay.
let's see what the, yeah, I guess we come up 10 meters or something like that, because it was 1065 when I walked in the room. But maybe the visibility's not gonna be good enough down here. I guess we could, do we think the cloud is us, or do we think this is?
That was probably just coming off the wall, but directly below me it looks clearer. That's
No,
just other clouds.
It's just super steep.
It's
That's
still
why we're seeing
steep,
the avalanche. That was you.
Yeah, because I'm backing up, because we can't see if we go face down, but backing up also throws the wash on fine sediments.
So those watching can see the challenges of working in a canyon full of soft sediment that's very easily resuspended and moves a lot. That occurs naturally also. So all the animals that live in here are pretty resistant to a lot of suspended sediment. We can continue moving away from this site, but just to get into clear water.
Don't go down at all. Just go straight back.
That's not that bad.
Do you want to run this part of the dive? Since they're your samples, would you like to run, to sit here?
No?
It's up to you if you like.
Do Seeps, I'm sitting in. Have to see.
Okay.
I'm sitting in the morning, I can see.
But just, I was thinking for your samples, if you wanted to decide,
Well, okay.
it's up to you.
Okay.
I don't mind to be here, but they're yours.
Before you come down to my
Well,
Yeah, either way,
okay.
it's okay. You want me to take them?
Yeah, because I have to make something.
Spend about 3 turns. Step back over.
Yeah, we left the outdoor cons after that. We're trying to blast all the dust off the sub so we can get dust. Visibility.
That was an Anipniastes pelagic sea cucumber that just flew by.
Sorry, I was checking to make sure, because I sent the email kind of late. Happy Day / Gracie are also hoping for 2.
They didn't tell me.
Oh, I put it in, they told me, or I talked to them after the meeting, so I put it in the email that I sent you.
Ooh, I missed that. Okay.
Yeah, sorry.
So we need 13.
Yes. Them and Jorge are both, if possible. We're hoping for like one that canyon for and then one more shallow. Like as shallow as we're going, but with the video transect, I wasn't sure if that was possible, so just wherever.
So they don't want 2 in, neither of them want 2 in the same place?
No. If possible, no. They were both hoping for one steep as possible and one for as shallow as possible, given the video transect.
But not including the 2 dives, because tomorrow is deeper. This could be the shallow one if tomorrow's dive is deeper, is that
Okay.
right?
Yeah, so maybe 2 today and 2 tomorrow would make more sense. See if,
Wait, oh,
yeah, 2 deep and 2 shallow.
2 for
One for each of them, yeah,
Okay,
if that makes sense.
okay. Yeah, I missed that email, sorry.
No worries, sorry.
Okay, we're back into clearer water. What are all these things floating above the bottom? Can we zoom in a little bit? Are they all small Anicniastes? Yep, wow. This is cucumber soup. A little soup of pelagic cucumbers. They look pretty small. And a rat tail, maybe? I'm not sure what fish that is. They're beautiful. These sea cucumbers tend to hang out near the bottom, but we've also seen them sitting on the bottom some of the time, so they may feed directly on seafloor animals as well. I'm not sure what they eat, though. Probably small particles. There's certainly a lot of them down here. Usually, most dives I've seen, they're kind of solo and one here and there, but I would say we're in a sea of them. Then there's a small jelly fish. I've forgotten the name of that one. We see it also off California. This version of an Anicniastes is very transparent, so you can see the coiled gut inside it, full of what looks like sediment particles or some kind of particles.
And so what is it their gut that we're seeing on the inside?
Yes, coiled gut. Intestines.
The jelly's just floated above our bio box. A couple.
Okay, I think there's also other small creatures in the water, transparent, whitish jellyfish, or maybe medusal stages of jellies. So we can go down when you are ready and try to sample the bottom. We'll want to, again, sort of scan the surface of the mud if we can.
Slope here.
Canyons tend to be quite productive systems, partly because the currents focus the food, sometimes trapping organic matter as well. So the presence of all these Nympniastes, pelagic sea cucumbers, probably represent the fact that there's a lot of food to eat here.
So that was still quite sloped there, so I'm gonna push this away for a little bit before I put it down again, so we don't disturb the side.
Okay, we're looking for a fairly flat part of the canyon floor to sample.
Right now it's flat, yeah. Okay, Mr. Paris, quite a bit flatter. He's on down. Would you rather have it down more slope a little bit? Maybe you should set that down anyways. There's the direction of the current.
It's interesting behavior. You can see the Nympniastes settle down on the bottom and then put its head end onto the sand. So perhaps they are feeding.
So it washes under us. Yeah. So it washes again behind, because that would, it's working with the coasters, just there.
It's a great collection of jellies here, jellyfish of various sorts. Again, we see some modeling on the sediment surface, and fecal casts tell us that there's life below mud, the surface of the mud. Also see a little bit of indication of sand wave.
Okay. It's just birds.
We'll take a little zoom in on the mud
Yep.
surface, please. So we see lots of fecal casts of deposit-feeding worms. These little piles of spaghetti are what spaghetti-like structure are what I'm referring to. So underneath each of these, there's some deposit-feeding animal. I don't know for sure that it's a worm. Could be something else. You see some of the bigger casts are probably a different species than the ones making small casts. They could be made by spionid polychaetes and subsurface sea cucumbers, echierans or sapunculans. There are burrow openings as well. They could have mollusks below. So we'll be push coring here, and we'd like 11 push cores. You happy with the spot? Hachi, you want to introduce yourself? Everybody else did their introductions. We have our co-lead here.
Gracias, Lisa. Mi nombre es Praxedes Muñoz, de la Universidad Católica del Norte. Vamos a tomar algunas muestras de sedimento en esta zona. Estamos cerca del eje del cañón. Este es uno de los más anchos del margen continental chileno, de aproximadamente 10 kilómetros. Las muestras van a ser analizadas para caracterizar los sedimentos y el agua intersticial o el agua de poro, analizando algunos elementos como sulfuro. Obviamente en esta zona no tenemos metano, pero se van a analizar otros elementos químicos, también de la fracción sólida. Los cañones en general son zonas que canalizan el material continental, así que también es una información muy importante para entender cuáles son las condiciones o características del sedimento continental que llega al fondo marino. Nos ayuda a hacer algunas interpretaciones estratigráficas en testigos o en sedimentos marinos. Nosotros llamamos testigos a aquellos muestras de sedimento que se toman con algunos tubos que se penetran en el sedimento, y eso nos permite ver la estratigrafía y ver registros que dan cuenta de las condiciones oceanográficas de las aguas del fondo.
Do we know what waypoint we're at?
We moved off waypoint 1. We're, looks like more than just over 100 meters to the west of waypoint 1. My understanding is the watch before did a video transect for 100 meters. Did you guys record the start and stops on those videos, on that video transect?
I was not there at that time, but I believe that
Could you check Sila? Is there a way to look and see if it's there?
I'll confirm.
Hopefully these sediments are nice and easy to core. Unlike the Seeps, which have subsurface carbonate, which sort of blocks the core, and sometimes it's very soft and jelly-like. I think these sediments should be reasonably well compacted and coreable.
I am not seeing it on CLOC, but I'm gonna confirm with the data loggers from the previous batch real quick.
Yeah, because we'll probably have to do another one if they didn't record start and stop.
I'm gonna check the
Okay. We can do it on the way back to waypoint 1.
That's the bottom right there.
How far in is it? Can you tell? Just halfway?
Roughly.
Hachi, is that going to be enough? They're halfway. Or do we want to look for a deeper place? It's okay? Okay, well, maybe the spot next to it will go up further. I wasn't expecting to have a rock, a heart a hard stop halfway down the core.
Yeah, it seems so pillowy, right? Seems like it's gonna be soft.
Should be. You never know. You never know what you're gonna find.
So we can make up for it.
So for the loggers, I'm pretty sure the Patricia, who's requested the video, also wants observations made along the transect of what's there. Yeah. In addition to recording the start and stop.
Sounds good.
Would you like the start and stop of your biology observation or elsewhere?
Sure. We just have to probably tell her where it is.
Okay.
That's it.
That's bottom, yeah.
Yeah. Wonder what's down there. Canyons generally accumulate a lot of sediment, so canyon floor, so there should be a core full of it. But so our water depth is just under 1,100 meters. We're at 1099, taking a series of 11 push cores for the various scientists on board. Eulogio Soto will be characterizing the benthic communities. Praxedes Bunos will be characterizing the geochemistry. We have, let's see. What will Davide be doing? Foraminifera? Yeah. Gracie and Davide will be looking at foraminifera, and what does Jorge look at?
I believe he's also looking at juvenile microbes.
Microbes.
Yeah.
Okay.
And you're on good all day. I guess I'll just start. Did you forget? Oh, I got it. Well, let's see.
Tyler, are you the artist who, on the basket maps?
What's that?
We- we-
A little picture, let me.
Oh, look at that, an eel. We admire your artwork each night. I don't know who else is drawing there.
Yes, I think Sunny tries to draw pictures sometimes. Yeah.
Okay, you guys, we enjoy it.
Yeah. So, you said nice one yesterday.
Yesterday's were magnificent. We thought we should frame that one, the hairy crab arm.
That was so. Nice.
Who is that? So-
Sophia.
Sophia.
It was awesome.
Yeah.
I forgot I did that.
Oh, I didn't look at it. It's Max Porch. Maximum Porch.
So these push cores, when they come back to the ship, there's a whole process involves taking away the outer quiver. They're actually in two quiver, an inside and an outer quiver. The inside quiver is removed with the core and put in the cold room till people can get to sectioning, and then they're eventually taken apart, put on extruders, and the mud is extruded centimeter by centimeter in many cases, sliced into vertical sections, and sometimes put in preservative like ethanol for studying the benthic animals, and sometimes frozen for geochemistry. The pore water is extracted, which is the water in between the sediment grains, for geochemical analyses. All different things happen to these sediments once they get on board here. This mud has a life after the canyon floor. Some of it. Or a death. You're gonna make a second row of them?
Yeah, we spun a little bit, so kind of.
So somebody can remind me, Greg would like us to slurp one of these little pelagic sea cucumbers after we're done with this. So Aaron, we're going to collect 11 cores, and rather than having 2 for Jorge today, one will be for Davide and one will be for Jorge. And
Sounds
then tomorrow we'll get the others.
Yeah, sounds good.
Basically the same thing tomorrow.
Thank you.
Keeping track of push core needs is one of the challenges on a ship when there's about 8 different laboratories and represented with different programs. I don't have any more space on my map to go to the left. I'm with you.
I'll give you a second page of paper.
It's between. we draw a paper map on board ship, even though we have the beautiful video of the core layout, we also draw them so that people can see which cores are next to which other cores. Often the scientists like to share information and use information from one lab group to help interpret the core data for another lab group. So knowing which cores are next to each other is important.
Which way? That's not secret. Right where you are? Pretty much. The other one just like this one. I might reach over for that one. Reach over. Push it a little bit. The camera's a little where it is now. Let's push in. So that's off. Okay. I'm happy. Yeah, I'm super. Yeah, I'm super happy. And I just that too. Yeah, both. Very content. Just aesthetic, super happy. I was like, very happy to see it. Alright, cameras in. Push out real easy.
1, 2, 3, 4, 5, 6, 7, 8, 9.
Real easy. Real nice.
6, 8, 10. So one more?
Yeah, this should be number 13 is number 11. Make it real easy. I might show it just behind you. Yep. That seems like a reasonable spot to me. Right here, here it is. Oh, because I grabbed it as deep enough, I can push it this way. Yeah, it looks like you want to pump these into this.
So that it's possible that, you know, what you're hitting on the bottom is some sort of stiff clay layer. I'm thinking if you push them a little bit into that, maybe it'll help plug everything for,
Oh, I see
keep
what
it
you're
in
saying.
on
So
the
when
way
we go to-
back.
When we go to grab it, just give it a little bit of a push.
Yeah, because the sediment looks pretty fluid otherwise.
Yeah.
And we don't want it all falling out. So if there is a stiffer layer in the bottom,
Try to
we
work
could
into it a little bit.
make like a little plug.
Yeah. So we want to do these ones at this time, and maybe some down first, and give this more, less chance of. Right.
So we should take a good picture. You guys get a frame grab of the layout there. looks like a little garden, a push course.
Nice spread, Alex. It was great because the RV's swinging around and giving me all the options.
Or
Change your perspective.
What?
Since we swung around and gave him, a wider field of.
Yeah. Well, if some alien came down and studied these, they could do a nearest neighbor analysis and see that they're territorial and they space themselves out.
Predatory
They
push
probably
course.
fight each other.
Yeah, what makes you happiest?
I'm thinking more about the border than anything, because the seven most challenging, so kind of either one pick the easiest or the hardest one to get back in.
Yeah, because if you could clear out like 9 and 14 and put them down here, then 7, you can just hit one of these corner ones. Does that make sense?
On the outside corners.
Yeah, so if you go these two guys, it's down here.
I was gonna load them from back to front.
But then you have the run risk of dropping 7 and then the other twos.
Okay, so I think front back.
Yeah.
The pilots are strategizing on the best way to collect these without disturbing the others or letting anything drop into the quivers
Unless it's the worst chance.
that's unwanted.
Just wanted to be better. It's gonna be the worst chance anyways. Yeah, 9%. Let's try that version. So let's see how that goes.
Yeah, so I can court you in. We rotating.
Sure looks like it. We're gonna have to figure it out because
I'm gonna spin against the I'm just gonna rotate us around nice and easy. Try to draw the fortune. No, we're not gonna do anything fast.
I like that it's rotating around the portrait. That's good.
So we're touched down here, so that's what
Somewhere on the edge. Oh, I forgot there was 8 over there.
Yeah, I forgot.
Don't leave 8 on the ground.
Yeah, they may wanna snap another picture then too, because that was off.
Oh, it wasn't in the picture. Okay, loggers, we need another photo because we think number 8 didn't make it into the family photo here.
Well, we put it that way.
Yeah. That's the marks go.
Okay, so we'll do.
Yeah.
6, 7.
Yeah.
Let's hope the sediments are stiff enough to stay in the tubes.
It's a great one to start on too, because it does check.
Oop, Well, we can try shaking it out, because we lost about half of it, I think. Can you tell it looks pretty clear now? That's what I was thinking. I mean, actually, the sediments look pretty coarse. Did you tell? Patchy,
So I can't push any harder than that, because I was lifting up the arrow.
well, if it turns out they're too sandy, we're not gonna be able to core here, but let's try it again and, if you find a place that's not disturbed.
So, more apples up there, right? But, it's over there. Yeah, you can also try the other ones that we can rotate and take a thing on the side. All come work that out first. The camera. What's this turn?
I mean, if they're fairly loose, I don't know if we can get a few of the closest ones into the closest
Just do it like
jiggers.
socially.
Um,
Well, that's what it's called.
I don't know. Well, let's see, because, yeah. Let's just see. Let's try a couple and see how they go.
So yeah, that could have been an option. So, whenever, what's this turn? Just try to- So that the motion. Sorry, struggles. Like,
He did.
perspective, like-
He did. He said he's lifting the ROV up like
So-
so they can't go in.
This is right all the way out. This is the best side. Yeah, that's.
He said he
yeah, no,
pushed
that's
this
fine.
far, the
Yeah,
hardest he
super
could.
good. Is that always in there too? Yeah, no, that's, yeah.
So we'll
Yeah,
have
so
to
that
see
should be.
if they're sandy.
As good as
It looked a little bit sandy.
So, I have so many bits of processing power.
So they have to
Does,
shake
I have
it
two
up.
bits. That's understandable.
Let's see. Let's try a few.
It's better than camera, not from the information I can tell and stuff.
Bueno, tenemos algunos problemas para obtener testigos de sedimentos. Aparentemente es muy arenoso, lo cual dificulta que el material se mantenga dentro del tubo, bajo en la base. Debería haber una arcilla más fina y más dura.
Yeah, oh my gosh. Yeah.
Bueno, esto es justamente lo que de alguna forma pasa en los cañones, que tenemos un sedimento que obviamente de origen mayormente continental, más antiguo, estas sedimentaciones son mayores que en la zona de plataforma, pero contienen material menos orgánico, más pesado, por así decirlo, esta partícula, que en las zonas de plataforma. Y por ello probablemente tenemos un sedimento más arenoso y menos compactado.
There's a lot on that trying to get in. That didn't produce any good. You know, just make good parts. No, it's hard to jam through. see what happens.
There are push cores that have core catchers in them, which are little one-way flaps or valves, but many scientists don't like to use them because they disturb the core as it goes into the sediment. It disturbs the edges of the core. They're usually made out of metal. Looks good.
Vantage point. That seemed to hold a little bit. Basically the location. Yeah. But that one I didn't put as much force, but just let it sit longer. Just the force on it. Maybe try on this too. Oh, I see. So like just a tiny bit of lift there. I don't know. Yeah. So just, yeah, that's good. That's all. I'm just kind of barely. Just
So I was hypothesizing these cores would be easier to take than in the bacterial mats at Seeps, but not necessarily true.
They're tricky.
They're tricky.
guess you're in control of whether it's in or like how much save you really have. Just that's your almost push down a bit. A little bit down. Nice and easy. This. Stack them up. Just a second. Looking off.
Lisa, after the course, we should do the transect you were suggesting. We don't have any logs so far for the start and end.
Okay, we'll slurp the Miastes first before. But then we'll use the transect to go probably back
Let's do that.
Sounds good?
I mean, it's
to the
not-
waypoints.
Oh, no, I'm not talking about the result. The part that I can. Yeah, it's, yeah, the part that you're in control of. That's good. That actually is a little bit worse.
Nice work. It is. This is tough.
These aren't fun ones. Yeah, that was it. Okay. Good luck grabbing it, you want me to flip it? As long as I don't have to re-index, it doesn't really matter. Oh, but there's a close one right in front of it right there. Something that I don't have to re-index them, okay.
So the the reason we push all the cores in first before collecting them is you can see when they're recovered, some sediment falls out the bottom, so this avoids disturbing the areas that we're going to core in advance of the coring. It's better the disturbance happens after the cores are all secure inside their tubes, the sediments.
Yeah, oh, I don't think that's been integrated in the video yet. That's fine, just go ahead and just do some links. It's enough for it. Push it out. Bust it down, bust it down. Nice and easy. Not spinning, that's not makes me happy. That's a pretty good still. That's a smooth movement. Movement, smooth. Still. It's also.
Lisa, a reminder in our request from Elohio, a reminder that we need a trip to Niskins over the pushcart area, and
We've done that before.
And Elohio was suggesting, since some of the sediment could be disrupted or falling out or land in a different sleeve, that we should take a couple extra here, since there's available push course space today.
Okay. So where, did he write that on email or where is he? Right here. You know here?
Yeah, that's probably what it is. Because that's the direction. We should have just done it. I didn't notice it. So,
Do you want the Niskin right here now that there's all this suspended sediment, or should we move away a little bit to cleaner water? That's one question. Yeah, but it's too late. You should have said it when we started touring.
40 feet, 45 minutes.
So go away a little bit for the Niskin. Okay, and then, do you want to push cores in here, or you want to move a little
Here,
away?
here.
Because the sediment's all disturbed now from this.
Maybe, but I have a comment. For example, this one, 14, I was talking to Gary. Perhaps it's not useful for anyone. So I prefer, because we have enough push core, to take more, like a backup.
Most of them, look, they'll all be like 14.
Yeah. Because what happens if we have, for example, some push core and it's not useful for another? I think it's
Then
better.
you take 5 instead of 8 on
Yes,
this day.
so for that reason, it's better to have more
You can have more, but make sure
for all. For all.
it's easy to have more, but leave some for surprises because you need to be able to sample something unexpected.
I know.
So how many, so we will,
In case
2 more, we leave.
like a backup.
Okay, but there's also, let's see, on the dive plan, is there another set of push cores on the dive plan or not? Section, animals. I should have put the Niskin first. Yeah.
Sebastián
My suggestion is
Eh,
to take
resuspende fácilmente la parte superficial y en estas zonas la tasa de sedimentación es muy baja, dada la profundidad que estamos a más de 1000 metros, por lo tanto se resuspende parte importante de material que es de épocas recientes. Entonces, la complementariedad que tiene de usar este tipo de sacatestigo de muestreo de sedimento con algunos otros que penetran más en los sedimentos son complementarios.
That's fine. Just back up for some. Slowly rotating us around, so it's. Yeah, it'll clear up.
They're out there.
It's actually coming out quite nicely.
Should redo it. More like ET phones home, but like. You're can't do it in the correct way. I don't know if it's easier to punch into more now or to continue correcting these.
It's really disturbed on this side of this course.
That's what I told you, yeah, but he said take them anyway.
You might go reach past it. You know what, actually, that's a good opportunity, we'll just do those with the board on. Let's think about that. Do that, so we'll just do those things else. Do you wanna grab these and then do those? Yeah. In case we can shift around. Yeah, okay, yes, we'll pull these 5. And we'll pull these 2. This is
So there is a few.
We'll just do a
Some are in numbers and
the challenges with so many push cores is that they're hard to reach with a single arm. So they're strategizing about how to get them, how to reach them all, and we've had a request to take a couple more for backup, as a few of these might be disturbed. And those probably will be taken with the other manipulator. So the pilots said they are happy to take 2 more cores, but they need to collect these first, the ones that are in the bottom.
That's good. Let's see.
And we have a visiting Anipniastes. Hello.
Oh, boy.
Goodbye.
I'm good, you good? Sparkling.
Cucumbers on the surface.
Sure.
Be salad in there.
No, why would you?
Sea cucumbers.
When scientists talk about benthopelagic coupling, they're talking about the exchange of food, energy, materials between the sea floor and the water column above. And organisms like these pelagic sea cucumbers that you see here are an important part of that. They appear to feed both in the water and on the sea floor. They deposit waste. Looks like I just saw some falling out of an Anipniastes onto the sea floor, so they move materials both up into the water column and down.
a very random fact about sea cucumbers, but when they're stressed, they're able to expel one of their whole organs. But I've never been able to see it. I was wondering if Lisa or Erin, yeah, I think it's called the Cuvarian organ, but I've never seen it live. I was wondering if any of you.
I've seen that
have seen it.
happen in the lab after they're collected, but not on the seafloor. Looks like one is unhappy down there. I wonder who eats the sea cucumbers. I haven't seen anything preying on them, but you'd think that the Humboldt squid might like them.
Oh, nice.
Yeah, you may have to pitch over to get these next ones. That's all elbow enough. So you had to rotate into that. Yeah. Movement so once you get it started, it'll hold the whole edge. They dump out so much that it's hard, right? Did you just do the lines? I just did the lines. Yeah, they touch it. They have to like a starter lower. Exactly. That looks better now.
More strategizing going on about how best to get these cores into the quivers successfully.
That's it. That's the one. Yeah. what I dumped behind it.
So that was 12?
Well, so I dumped that one behind it, I missed it,
Yeah,
so it's empty. So I can put that in with those 3 if you'd love. So we still have the 12.
sure. I mean, if it's easier to repunch it now, you can, or you can put it, hold,
Okay. Yeah, just.
or put it back in the quiver and
just
do it
find
later,
else.
whatever can be easier.
Put those 2 away, do some over there. But yeah, I mean, it's so close.
Well, I overshot it by like more than I wanted to.
It's not like the time, it's 2 seconds. It's more than how much last.
Well, it has been.
I've been watching. It's a lot, Jerry.
He sees it when I slide the shoulder on the stone. Honestly, that's because it wasn't supposed to come out before
Seems like the sediment's getting slipperier.
There's not much time before it. They want more checkers. One over here.
So it might be sometime in the sleep.
I wonder if if there's any heterogeneity in the mud, if we turned slightly, whether we would get anything better, worse, I don't know.
I can do it, we can check. Pivot to the right, so it's not.
Because that particular spot looks really difficult, just where 9 and 12 have been.
Yeah.
I mean, they keep falling out, so I'm not confident that if we keep trying that, but I mean, we can be a quarter meter over to the side or back or something, right? I don't know if it'll make a difference.
I can just push it up to the right a little bit. If that works.
I mean, you know, the 7 and 8 seemed okay. You got them in. I don't know if the med's any different where they were over on the right side.
Try to keep my reachable footage there. That's good. Alright, let's see down here. Let's just. Let me sell our real quick. It's us. Planted. Planted like a push ball.
No, but I have like
Taking push cores and unconsolidated sediments is really pretty difficult. This is the part of the process that never shows up when the scientists publish their papers. You just see the beautiful data that's generated by the cores. And in the acknowledgements, we always thank the pilots for their efforts, but this illustrates, you know, the skill required and how frustrating and challenging it can be. Has anybody identified those fish? You guys know what they are? Can they
we
be
can look them up.
eel pouts? I don't know. The fish?
I think Maritza sent a fisheries guide from Chile yesterday, so I can look it up. We didn't have any good screenshots of the fish.
Look up eel pout and see if, in the guide, and see if there's anything there. frustrating.
No, it's not so, I think it's just like, for me, my motor skills like degrade as an,
As you go.
yeah, as I go, so.
Well, it's challenging sediment.
Tyler's gonna make it look easy.
I don't think so.
Let me know what you want for please.
This is 9, right, that we're moving around. Are we over to the right of, wait,
Yeah, we're away from
to the right of where 7 and 8 were originally, of the far end?
the, we're more like outboard, like.
Beyond
yeah,
it?
Yeah. We can go to the right if you see this.
No, it doesn't matter, just go wherever. It's easy, I just need to map it. I see what you do. No, that's cute.
Maybe that's what happened to the other one, did not stick. Yeah, so that area is like,
They're both.
yeah, they didn't stick.
So Pachi, can we move a little?
Yes.
After we collect these 2 on the bottom, can we move a little bit and try another spot? The spots.
Do we want the Niskin bottles here over the fridge first?
Well,
Later?
I'm gonna suggest we move a bit.
Okay.
And try to take that, we need these 2 and 2 more, so we need 4. I mean, you could put these 2 back
Yeah.
and then use the other arm and just take 4 in the front row, maybe? No, we can't empty the tubes because the mud's in the bottom already. It would destroy anything. Yeah. So we can only empty tubes that are out. Yeah. So, yeah. It's just, I think it's better to be a couple meters away and have good cores than to spend the whole dive here trying to sample and not getting anything. We can try a little bit, another spot. I mean, if people want, they can try to multi-core here, maybe. It'll be easier even. I mean, we can take a Niskin. Yeah. The next spot. So I think we just move a bit. And then take a Niskin before we do the next set.
It's like a couple meters.
Yeah.
So the current is coming towards us, we're moving up, change the current a little bit.
Yeah, but it blow past us, so it's clear, take a Niskin, and then let's try to punch for quarters.
In that spot?
Yeah.
Okay, so just coming, it's not like 100 meters?
Right.
Okay.
Just so that they're near each other, they're near enough to this round. Okay, so that was 9 and 12 that we did not take, right? Yeah.
Appreciate it too. So we're gonna go ahead a couple meters, fire Niskin, and then do the porch. Okey-dokey. I'm tempted to leave the porch out so I don't have to scoot it. See that it's another vegetable when we come down, so touch it down. Yes, touch it down. Yeah, you will. Push it forward. That's gotta be,
This sediment is unconsolidated, has a high water content. It's one of the things that makes it fall, but it might also be coarse-grained. You can't really tell. Coarse sediments don't stay in the tubes very well.
what?
I'm used to seeing this problem at methane seeps, but I'm surprised that we have it here.
Doesn't need to be out very far to get that first row.
It's probably far enough, yeah. So we're just moving to a slightly different spot in hopes the mud might be a little more cooperative and that we can get some undisturbed surfaces. We're going to take a Niskin sample for geochemistry in the water above this next patch of cores. All right, let's just see, is this clear enough? We moved, but there's still particles in the water. It's okay? Okay, do you want 2 Niskins? Because number 1 has been failing consistently. So please fire 1 and 2. Yeah, for whatever reason, 1 hasn't closed on properly in the last 2 dives.
That's good to know.
You should.
Yeah, that's a little bit today. You don't have a automatic on, so you're sort of able to spend. Alright, so oh, okay. Alright, this thing's coming down. That's 1. That's 2. Could just make it a little this one. Yeah, they took that one out last time.
Apparently one of the lids has come up at 45-degree angle, and so nobody knows if it mixed with the surface water, so we haven't been able to use it.
Yeah, fair enough. I know they made some adjustments last night to try to rectify that spring action. That was on our visual feedback 2 Niskins fired.
Mhm.
They flagged under goodness.
So you guys got that? Loggers?
Is that the same one? Yeah.
1 and 2. Yes, because patchy 1 has not closed properly for the last 2 nights. I didn't get my sample yesterday from there.
You don't want it fired.
So we have to try to
And
then get,
we can see that one did not fire correctly again.
is there anything we can do about it from here?
No, I can't reach it. Can't locate it either.
Okay, so you can write number 1 failed, probably good to put that in the log. Okay. I mean, do you want a third one? I think, do you want a third core? Third Niskin? Number 1 is bad.
Yeah, if possible.
If nobody else needs them, I mean, Anna probably wants them, but maybe we'll fire number 3, just to have another backup.
That's where 4 wants to go. Four started moving, so we're past 3.
Yeah, because it'll get closed, but the cartridge didn't come out.
But no.
Oh, okay, so, no, so 3, we can see that 3 tripped, but the indicator didn't get, but you can see the flag hanging on it. So that's okay.
3 is fired.
Yeah, it's fired.
Thank you. That's weird, why wouldn't the visual trip move? I don't know.
Well, I don't know.
All right, port arm coming live.
So we have another arm getting into the action to reach push cores 1, 2, 3, and 4. Hopefully the mud will be a little more cooperative right here.
Just line up with your jaws.
Yeah. That's the spot. What a guy, what a pal, what a friend.
You're gonna be able to see, I guess you're just gonna have to align your sit. This is really soft stuff. That's good. Be able to push through something.
Yeah, we might have a plug on these finally. That's picking the sub up. Let's go.
They're like half full?
Yeah, we hit bottom pretty quick, but we're able to push in a little bit further. Do you want me to try, see what this looks like with the other 3 in?
Sure. Let's see if we can get this one in.
The current is coming across.
So, yeah.
Should have plenty of space.
Just, we'll test it, see if the spot works before we take the other ones.
We'll do what you want for the camera. You'll pull out three on the sides. Yeah. Just kind of push down a little bit. Drop it. Let's just let them settle.
Okay, and then ease this back down. Thrash around. Okay, good.
Get a little perspective of your arm, since you can't see the quiver. Yeah.
Alright, coming up. Want it in?
Yeah.
Okay.
Oh, well, if we lost some of it, it's hard to say. It looks pretty short. Looks like it's only about 5 centimeters. So did some of it come out the bottom? I couldn't tell. Yeah.
I was hoping we got the flow because it felt like we went through something. When you rock, you can break a flow up sometimes.
So Pachi, do you want us to take 3 more here? Is this gonna work okay?
That's definitely
Right. Yeah, that's just handy.
That's what it's super simple. It's all just considered, so it's really good. Really good job.
Thank you.
Yeah.
Sometimes can't fight the sediment.
I feel like I thought it. You know what I mean?
I strike rate.
It's not moving.
I know,
Well, the alternative was like go slow, lose all of them,
yeah.
or go fast and lose like 80% of them.
Yeah. Yeah. It's not a good spot. If there's somewhere
Well,
else.
this is
Yeah,
the
should've said that.
characteristic of the axis of every canyon, the currents come down the middle. So, if you want the samples in the bottom of the canyon, and we're near the slope also. So is this layer right in front of me, stuff that came out of our core, so we don't wanna core over that?
Yeah.
Okay,
We're
we
just-
wanna just try to find a spot where we haven't disturbed it.
We're just looking at the texture, it's just.
It's sandy.
Yeah.
But fortunately, it's all down current, so there's plenty of big areas to.
Yeah. So one of the things we're seeing is that the sediments are a little bit coarse-grained. Looks like some sand-sized particles fell out of the bottom of that last core. And that's a product of strong currents in canyons, which is pretty characteristic. We didn't really know what the sediments would be like here, but I'm guessing we shouldn't be surprised to find fine sediments with coarser sediments below. We're gonna try to get 3 more cores here.
Corrientes de fondo son bastante fuertes.
There's clearly a really flocculent layer with high water content at the top of these sediments and a coarser sand layer below, and that's probably what we're running into that feels like a harder bottom. Okay, we need to keep it as upright as possible.
Yeah, we're trying to.
I know.
Get some down to us that we get through, because it's spotted pretty real fast.
Bueno, esto como es una característica de los cañones, las partículas más finas son
That bottom is
transportadas,
probably just that sandy layer.
por lo tanto también hace que los sedimentos sean más arenosos
Easily penetrated by push cores.
y está costando poder hacer entrar los tubos en el sedimento más arenoso, aparentemente una capa abajo que está mucho más compactada.
It's not even strong, the sediment's not even strong enough to hold the core upright.
There's nothing you guys can do with this, you just get frustrated over it. Just keep trying until we stop trying.
Thank you, guys. Sorry it was so tough.
Tomorrow, they're gonna be great tomorrow.
The 2,000 meters should be maybe easier.
Bueno, asumo de que hay poca materia orgánica o al menos menos que en otros
So we just
sedimentos
have a
que los hace más compactados.
What would you like to do with this? Because, struggling to even get it to stand up to get back here.
You want to shake it out, actually, or you want to try to collect it?
Oh, we try. Oh.
You can shake it out I mean, you can pick it up and see if any of the mud stays inside, but I think it won't.
There's nothing gonna
Yeah.
stand.
Let's shake it out.
Oh, it's not so bad.
That's just the side.
Yeah, just keep it.
Just shake this out.
Okay. It's gone. Pueden ver cómo las corrientes se llevan rápidamente el material
It's probably sand. I think there's just a layer of sand down there. See if we can.
Either way, it's making it already up, so I can't get any deeper.
Well, let's just try to collect and see what happens. Oh, it's a bad fish.
Go away, you sloppy sucker.
It's in for the kill.
Alright, so
He knows
watch
you.
out.
It's all over there.
Watch out.
It's all over there.
Okay. That camera out all the way.
Thank you. I tried.
We'll try two more.
Margarita.
Sounds good.
As far as it'll go.
That's as far as we're gonna.
Does anybody understand what that last outdoor message is?
I think he meant to avoid any disturbance on the sediment. He's proposing we use the 10 centimeter as a margin, so we can measure that we're far enough away.
Okay. So we have a request for suctioning one of the little sea cucumbers. We have a lot of mud on the porch that possibly will. So we want, I'm not sure whether we're seeing the big ones and the little ones, but I like these little ones because of the. You can go into, probably, should go to 5, 6, 7, or 8. So 6 right now.
Sure, great. You can find it. Got it. Did it,
Yeah.
it's in there?
Yeah.
Yeah. Wonder if we should get one more. Just to be sure that,
It's in there.
yeah,
You want another one?
well, maybe one more, I think. case it's a new species or something, it'll be a second one. If we can, we'll see. They seem more abundant near the bottom, but I know it's harder to see down there.
Yeah, it's fine. Coming up.
Doesn't bloody down. Just gonna let you settle out before trying to grab this thing.
Okay, there's the second one. So that's in number 6.
Yeah, 10.
Thank you. So 12, 13, 11. So that, we got 2 of them. Okay. If we can go back to what's marked as station there and then do a 100-meter transect back to waypoint 1, please.
Okay.
Was there anything else?
Get that rail.
You guys were putting in requests back there that we needed to do here?
The water transect is the only thing left, I think.
Yeah. We'll do a 100-meter video transect along the axis where we mark the start and stop. So maybe, and I guess you guys just stay a fixed distance off the bottom for these. And I think is there a speed like 1.5 knots? Something that you usually do for video transects?
Yeah.
Is there a sample in there?
Yes.
Yeah.
Mm. Didn't get marked down on the pilot. This. That sheet,
Okay.
are there samples of these two? This is blank, so nobody wrote it down. What's the second one? Nothing? Is there a sheet?
Lisa, did we start the transect, or can you let us know when we? Did we start the transect?
No, we have to be on the bottom And we'll be transecting along the bottom at a fixed distance and speed. And they'll tell us when we're ready to start it, right? So,
Thank you.
this is new for this group. We haven't done video transects before, so, on this expedition. These transects will be to quantify the fauna in a way that can be compared to the canyons to the south of here that will be visited by the next leg.
Yeah.
I'd also be fine with doing a transect from where we are back to waypoint 1. We don't have to go over to the station since we disturbed so much over there. Let's just see if we can find a spot on the bottom that's with good visibility. While we're doing this, let's have our loggers tell people about what it's like to live on the Falcor 2.
Yeah, I think the word would be spoiled. This vessel has been amazing. The food is great. We have like daily buffet style meals, which is more than I eat when I'm at home. It's very exciting, the fact that we get to do science almost 24/7. And yeah, Erin, what's your take?
I would agree. This is such a nice vessel. Along with with any expedition, the added bonus of there's always something happening.
It's very exciting.
Either I'm working on processing our own samples or planning, our future work for my group, or if I don't have anything to be doing on that rare occasion, I can find out what the mappers are doing. I can see what the animal people have collected. There's never a boring moment, for sure.
That's true. Do you think you've gotten settled down into a routine on the vessel?
Yeah, I would say so. How about you?
Yeah, I like the so.
Yeah.
Especially with the 12-hour dives, we get a chance to process the samples
Yeah.
in a timely manner.
It was also my first research cruise where I've had data logging shifts,
Awesome.
and I've absolutely loved it. It's some of the best parts of the cruise so far has been spent in this control room.
I feel you get to learn so much as well.
Yeah.
Like, especially species-wise.
Yes, as a micro person, I'm learning a lot.
Way bigger than your...
Any Donkey Kong tournaments yet this expedition?
What is that?
Donkey Kong? Donkey Kong is a video game, and no, not to my knowledge.
Not yet.
Not yet.
Bom dia.
Bom dia. Obrigado.
Okay, we're gonna start the transect loggers, but don't push start yet till we say, but we're just getting ready to do that.
Got an updated waypoint?
Sweet ass, let's go there.
Oh my gosh.
Yeah, so Nazmi find was 3.19.
Okay, are we ready now?
Yeah.
Okay, start transect. And then how do we know when we've done 100 meters?
Could you measure how far away I am from waypoint 1? Say every
Waypoint 1 is 111 meters.
Okay, we'll just go to waypoint 1,
Okay.
and when we're right before it, we'll end the transect.
Okay.
So we can't sample, but we do want observing. The loggers should record what they see in the way of bottom and pelagic fauna, I think. Don't know. So we're clearly on a slope.
Yeah.
It's a rat tail, grenadier or macrurid. We have the big species of shrimp, of which I never figured out the name. Same one we saw at El Quisco. We have an Hypniastes in abundance.
Yeah.
Yeah, that's a little better. We have a mottled seafloor, probably lots of fecal casts down there. Can you tell what the slope is? Is it like 20 degrees or 15 degrees?
I don't know if the answer you can tell me that.
It might be able to, Jules. I think the M3 would be able to give us an idea of what the slope angle is.
Yeah. It won't give us an actual
There's bigger fish.
angle, but we can
Just rough
break out our
definition
protractor.
or something
Yeah,
rough.
exactly.
I'm just curious if there was an easy way, but we
Yeah,
don't have
no.
to.
We can do that.
CQ was done before.
No, 6.
6, okay.
Our market or.
Okay, cool. Sorry, I'm just trying to catch up.
So right below us, I would say it's probably about 45 or 50.
Really? That's steep?
It's coming down now. So probably like 30 here.
Much steeper than I expected. And it's amazing, the sediment sticks.
Estamos recorriendo.
Tiene 129 kilómetros de largo. Aproximadamente 10 kilómetros es su ancho promedio y una profundidad promedio de 870 metros.
Do you still want the M3 on, or are you?
No, we're good.
Okay.
Looks like some trawl marks here, or could those be ours?
I don't know. I
Could be
wasn't paying
ours.
attention. They slid down the hill.
We have been here before.
Yeah, okay.
That was cool. Wait, just crossed what previous.
El gradiente del talud es aproximadamente 5 grados.
Acids in the water.
Yeah, I would say it's about 40 degrees.
And you guys just mark down the slopes, 30 to 40 degrees.
Is it
This transect.
angled? Are we rolling?
Pero aproximadamente
Degree and a half.
en promedio
Actually, the video transect also needs to go on your log.
Mhm.
you guys record that, please? Thank you. So we have pretty sandy floor here. I mean, we're not at the exact floor of the canyon, but okay, there's a white tube that could be a Hyolinicia or quill worm. No, we can't stop. We have to keep going. We aren't allowed to stop, look, zoom, or anything for these transects. I'm just telling the loggers things they could put down. I think we're supposed to go at 1.5 knots, but can you tell how what is our speed?
0.3 meters a second.
I don't know how to turn that into knots.
No, I don't know the knots.
Excuse me?
0.3 meters a second. How much knots is that?
Just double it, so it's 0.6.
That's fine.
4 is
0.8.
Yeah, that's fine. I think slow is good, actually.
We're almost there to
Oh, there is some sort of cnidarian. There's a little more animal life here. Okay, I think end video transect 1. Oh, that's great. So this is a waypoint where you have, I guess some collection was done already on the front end of the dive. And we have a series of waypoints that move us up and across, zigzagging up the margin. But let's just settle down and see what's on the bottom here, since there's a lot. I guess these were seen before. These look like heteropolypus or something related to that. I don't know if they've been collected before. Okay, let me just ask if there's any interest in these.
Lisa, can you expand on the subtype for the observation of the hydrophores?
I don't know exactly. They're a cnidarian. I don't think they're panachulids. There's so many arms here.
It's also jubilated onto the
Okay, good.
Dalton buffalo.
But I need some guidance from the Chileans and maybe Greg about which organisms they would like to see collected in this canyon. My understanding is nobody's had an ROV or a submersible down in this canyon before, and it hasn't been mapped until last night. So we're maybe the first people looking at some of these creatures. And I don't know how widespread species like this is, whether we're likely to find it in all the canyons on the slope. Maybe we could just zoom in and get some really nice imagery. See if anything's living in amongst the polyps here, or if they're very clean. pretty clean, certainly compared to tube worms, which are covered with animals. There's some burrows underneath. Okay, I'm hearing that we would like to collect a little one of these. So perhaps, I'm sorry, one of these two little ones over here. Again, let's zoom in and get a really nice picture of it before it gets collected. What, is this gonna be easiest in a suction or in a coral quiver?
Yeah, coral quiver.
What is the creature over to the right also? That's pretty interesting. You guys see the little pile of oval pellets? Those are fecal pellets, like that might be made by a capitellid polychaete or another deposit feeder. There's a great collection of different sediment pellets on the surface here. They're spaghetti ones. Spaghetti-like ones are made often by spionid polychaetes. Gives us a clue of how much life there is under the sediment surface when we see all these fecal casts and pellets at the surface.
Only coral quiver one video so far. Only one has been used so far.
As best as I can tell,
Yeah.
number 2 had something in it and crossed out on hers. These were taken before my watch and nobody wrote anything down, so.
Okay.
Loggers, do you guys have a basket map that shows what coral quivers are free?
Everything but one.
Okay, great,
So
thank you.
race between 2 or 5, 6.
So because this canyon hasn't really been explored or sampled much, there's interest in collecting for the Chilean museums representative biota. I'm not sure what kind of, they may have a trunk that extends down into the mud, and I just don't know how far that goes. No. This is going in number 2? 5, sorry. 5, yeah. $13.34. There it goes. Great. Fine. Great, thank you. Could we zoom in on what might be an anemone covered with forams, but it might be something else? So these are arborescent foraminifera living on top of this sea creature, which could be an anemone. I can't really tell. Arborescent foraminifera are protozoa that agglutinate sediment in little tree-like structures. We saw a lot of them at Seeps as well. They seem to like to be up above the seafloor on surfaces, so they'll attach to all different kinds of organisms. This is strange. I don't really know what this animal is. Possibly an anemone, but well camouflaged. We've seen sea urchins also in different deep areas of the deep sea that adorn themselves with foraminifera on the surface. Okay, thank you. It's strange. Maybe, I don't think it's a sea cucumber. Somebody said they want a yellow one of those heteropolypus, but I don't see any yellow ones. So I'm not sure what that comment. Anybody know in the logger realm what the yellow one meant? No. Okay.
Something like, it almost looked like the substrate underneath one of the heteropolypus, but I don't think it was yellow. I think it was like a whitish. It looked almost like a sponge.
Well, okay. Could we pan back over to the red creatures and see if there's something underneath them? Somebody was asking for something.
There's something strange.
Okay, maybe zoom in over here. I'm not sure what they were looking at.
Sponge.
Where? Oh, this
Yeah.
sponge? Okay, whoever wrote a yellow one too, who was that? Anna. Do you know, can you tell us what you're talking about in terms of a yellow thing? Was it the sponge you wanted? I suspect not. Get a little what?
So the one, you see the, can I,
Yes.
this one? Close to the one that you just collected, this one.
That's
Now
a
it's orange.
it's a sea anemone.
Yeah,
Okay,
he
you
wants
want
a sea anemone for colleagues, but in the other screen was yellow, sorry.
okay. Is that something we can section? What do you think?
Thank you.
Yeah, give it a shot.
It might be a
What jaw do you want to do?
one of the big ones. 7, 8, or 5. It may have a trunk that extends down into the sediment. I'm not sure about that type of anemone. We'll see. It does look small. What sampler are we on?
5.
5.
Which one do you want it in?
5 is fine.
5. Let me know when you're ready for suction. Suction on.
It does have a trunk that's anchors it down in the mud. I don't know how long they can be long. What we don't want it to do is clog this section. Ugh.
Put it in our Bible.
Hmm.
Yeah, too big for that.
You want to put it in our Bible boxes then? Do you want to put it in our Bible boxes?
Sure,
then?
if there's a way to get it there and out of the suction.
Yeah. Which Bible do you want?
They're all free, whatever's easiest for you. Problem is these guys are sticky.
Okay, kill it in suction.
Yeah.
Suction
Oh, it's going.
Yep.
There it goes. Okay, so that went in 4B. That's some sort of an anemone.
Yeah. That's the only downfall is retracted really on.
Okay, can we just poke that creature, this one, just to see if it responds?
With the
And
suction nozzle, or do you want?
yeah, with the suction off.
Okay, sure.
I don't want to suck it up and clog the thing. I just want to see what type of animal that is. Can we zoom in and see if there's a response? Yeah. I wonder, it's not a, I don't know what it, I'm wondering if it's a type of urchin, but I don't think. Does it feel soft at the end of that poke?
Somewhat.
Yeah.
Okay.
Yeah,
I wonder if we should collect it since it's so strange. You think it could be collected by manipulator?
Probably.
Why don't we put it in the bio box with the sea anemone? I don't think anemones will swim out, so they're pretty safely in there. I'm just curious as to what it is. And I always love animals that adorn themselves with foraminifera. My favorite. Ooh, I was hoping Greg would just tell us what it was, but he hasn't yet, so. Well, I thought it was an anemone, but it doesn't respond like an anemone when you poke it, so. And if it's soft, hopefully the manipulator can not squish it.
yeah, we don't.
A what? I don't think, I don't know. I want to say it's in the, more like in the echinoderm realm, but maybe not. Might be a type of sea cucumber that's round.
That's
Greg answered.
What did he say? Oh, if Greg's not sure, I feel a little better about this.
It was a good call. Good example.
Very nice. This is one of the fun parts of our job is sometimes we get things we don't know what they are. It does look like, it looks like a, oh gosh. So maybe it is a sea anemone. We'll go ahead and take it. It's got the scaly appearance of, yeah. It's got a lot of forams on it.
Greg confirmed anonymity.
Okay, yeah. So this will be anemone number 2. Can you write in the notes that it's adorned with arborescent foraminifera, please? Yeah.
That freaked me out. It's like, where is that coming out of?
Yeah.
It was already free.
Great, thank you.
Yeah. Anything else you want to see here?
Just take a look at what the white,
Can I see it?
is that a fish or a tube? Or a worm? Or a baby hagfish? Baby hag, little hagfish, right? No,
No?
oh, sea cucumber. It's a small sea cucumber. Let's photograph it for a second and see if anybody. Okay, I'm seeing would be good, please. So, do you think this is sectionable?
Probably. Far away, so you're gonna have to move.
What is it? Sorry. Okay. Yeah.
Yeah. You're gonna have to move a little bit. That's
Yeah.
not, you wanna kick the rail out on this section of
It should be pretty small, right? Like a centimeter across or something like that.
All right, let me move that a little bit.
Get there, so,
You think?
yeah,
Okay.
look at it.
It's not going to swim away from you, probably, which is good. And like the
You want it in
Neastes.
5?
Sure.
Okay. Suction on.
We also have to know the birds. Are we going to be able to distinguish them?
We have something in 5?
Oh, it's 6. Careful, I think it's 6.
Yeah. I think it's. I don't see it come through. Might be sticky.
Just shake your booze up a little bit. Stand it up and shake it. Always been trying to put any crowds up.
Yeah, push me away from this wall.
Alright.
There we go.
Yeah, like when to the right.
Did anybody see it come through at all? It's pretty small, so.
I didn't see it.
Turn the suction on for a second.
Off.
Just don't know if it's sitting on the top there. You see something sitting at the top.
Of the canister?
Yeah.
So that could be
I don't
it.
know what that is then.
They're sticky and it's small.
Try and turn the suction on.
Oh,
You see
yeah,
how it
that's
disappears?
yeah. Yeah, that's probably
Stuck
one
to
top.
the top of the jar.
Yeah. How's that
Oh,
one?
there, that,
Yeah.
that little white thing.
Yeah.
Yeah, that might be it. Okay,
Alright, let me-
there it goes. Just fell down. Okay, we're good. Thank you.
Index it.
Okay, let's head over to the waypoint 2. Along the bottom, and we'll just sort of take a look as we go.
On the sit down.
It's interesting, we saw that little sea cucumber, but I haven't seen a lot of big sea cucumbers on the sediment surface yet. I don't know where they are. Of course, we have the pelagic sea cucumbers, which are very dominant out here, so cucumbers rule along with all the cnidarians. The sponge, which we saw, is sort of hidden under a sediment, which is interesting.
Okay. You okay to move on?
Yep. How far is waypoint 2?
Right.
160 meters away.
Okay, we'll just take a look at the seafloor. So we're moving along what's still probably the canyon axis, not the central, but the edge of the axis to the slope where it becomes much steeper. And we'll start a series of video transects after that.
Bridge ROV, got an updated waypoint.
Copy that, thanks.
Should get a little bit shallower. Again, lots and lots of fecal casts down on the sediment surface is what's making the surface look mottled and two-tone. Sometimes canyons have methane seeps, often at the base of a steep scarp. There's a contingent on board the ship that's kind of hoping we might find some seeps here, but we haven't seen evidence of that. So rat tail. Our depth is 1079 meters. We realize the depth isn't projected on the YouTube live stream, as far as I know. So people, it is. Oh, I guess on some of it is, just not the video.
Is all the texture on the sand and corals from different organisms? And the different texture on the sand, those burrows from
Fecal casts
Fecal cast.
and burrows, but lots of fecal casts. In some places, there's a lot of surface animals making tracks and traces across the top of the sediment, but we're not really seeing too much of that here. Relatively few animals above ground. Shrimp, for sure. But we don't have the large numbers of shrimp that we had near the methane seeps at El Quisco.
The same size, you'll keep the same distance.
Probably were much deeper, so that might be one reason. Oop. Some more heteropolypus. Ooh, and can we stop octopus? You take a little look.
Well, we've been trailing down, this little trail.
Oh yeah, there's, just as I said, we didn't see any tracks and trails, we got one. It's a small octopus. Maybe a different kind than we were seeing in previous dives. Not sure. Could be the same kind, actually.
This is following us up the hill.
Where'd he go?
He's right in front of us. The visit's following us.
White out. Tan out.
How long do you want to wait?
Is there any current at all here?
Not really, just sitting right in front of us.
Well, we can keep moving. But you probably can't see where you're going, so.
No, once we move ahead, I'll get ahead of the this.
There's all the Idarians. Can you pan left a little? I just want to see if we're in like a huge garden of them.
I think it's it.
There's more. This is interesting. So this is almost like a giant field of these things. There were some below us and some to the left. And it would be a good,
You still have to stop these.
Yeah, stopping that.
Cheers.
let's just explore whether they're forming a habitat type. If these were, I don't think we call these corals. There's a lot of them.
Is this what we collected earlier?
It is. So we're not going to collect any, but I do want to see how they're distributed and if they form like a field, a heteropolypus field. There's some, I guess, hard substrate that they're attached to. So this depth is 1065. There's really a lot of them. Now we're getting into some other things. Okay, can we slow down and pan this? Take a look here. These clams are probably a cesta. They're really common canyon big white clams. They don't indicate a methane seep, unfortunately, but they are a common feature of canyon walls. And they're kind of big. As far as the deep sea goes, they're large. Oh, Greg says they're brachiopods, not acista. So let's zoom in and look at them. Brachiopods have a lophophore structure that makes them their own phylum. Well, there's a few groups with lophophores. So these are brachiopods. Greg, do you know which brachiopods they are? We'd like to collect, we have a request to collect a few of them. What do you think the best way to collect them is?
Probably using the scoop.
And then pour them into the
And then pour 'em
bio
into a
box?
bio box, yeah. That's
Okay.
probably our best bet.
Okay. So the brachiopods are the bivalves that they're not bivalves. They look like bivalves. They have two shells, but they're their own phylum. They're common in the deep sea. There are a number of different forms. We're going to try to scoop some of them off the wall. Greg, are we sure that the cnidarians are heteropolypus, or could they be another genus?
It's been unfortunate, but all right.
Okay.
How tightly attached are they? It's hard to tell. There's a couple. Looks like you got them. Okay, Greg says, can we zoom in on the one to the left in there that's open to see the lophophore? Can we zoom it and then, I assume he's talking about one we collected, but no, maybe not. Maybe that's what he meant, yes. So if we look inside the valve, the lophophore is kind of a U-shaped filtration structure that's visible, barely visible there. Okay, thank you. I don't know if that's the one he was talking about. So the biobox, it could go in 4A would be fine. Can you tell how many we got? 2 or more?
No, just let me see that.
Can we zoom in? Well, there's one for sure. I thought you got a couple of them. So maybe there's one underneath there. Yeah. So these are bracket guys? For 4A. No, it's a wall, a canyon wall, sedimented wall.
So,
I'm gonna try,
yeah.
they're still there.
Yeah, it's on.
Down now.
Yeah. Just those some stuff.
Maybe we'll try for another one.
Yeah. Oh, that'd be really good news.
That's helpful. Yes.
What did we get?
Diverse.
We got another heteropolypus. That's okay, we'll take it. I think, we don't have. Is there a brachiopod in there, you think?
In there somewhere. That's what I was actually shooting for.
I know. I feel like there should be one in there, shouldn't there? Down in the bottom. Because if we don't think there's one in there, we might leave this on the bottom. I don't know that we have so much storage space for too many animals, but
Can't say for sure.
okay, maybe oh, there it is, there it is,
Okay.
yep,
Now I can say for sure.
yep. The big pink cnidarians could be Heteropolypus or Anthomastus. I'm not sure if the name Anthomastus was changed to Heteropolypus or there's a new genus that's separate. Okay, that's good, thank you.
So yeah, I'm the rest of this.
So there's times 2 plus.
Be able to be in there.
Yeah.
Think it's gonna stay.
Yeah.
Sorry about the unneeded creature.
It's okay. There's quite a number of them here.
Got that marked down.
You guys have a few good shots of this wall with all the pictures. It's great. Okay. We will continue slowly towards waypoint 2.
It's gonna satisfy anybody's curiosity. He's gonna do it anyway when he walks in here.
Oh, there's a yellow coral. I wonder, I'll probably be interested. But I'm guessing we'll see a lot more. Can we zoom out just a little bit so we get a bigger view of it? Yeah, you go. So there's a lot of these Heteropolypus. They seem to form a zone here. We'll have to wonder if they're all separate individuals or if they butted off the same one. They are tracking the hard substrate that's available, and we can see the brachiopods all along the edges. Look at that. Beautiful. Wonder if we can get some sort of beauty shot of the wall with and there's an octocoral probably. Let's ask Anna if she let's take a picture of these and see if she wants them.
Moving on.
That's nice. Beautiful. Our coral people are saying it could be a plexaurid or acanthogorgia. That's a galathea crab on the coral. And there's brittle stars on the coral too. Over on the left one. It's interesting, one has crabs and crab and one has brittle stars, but they're not mixing. They each have their own commensals. Maybe the crab is scared off all the brittle stars. It's kind of neat. So the corals are a great way for these animals to get up off the bottom, these commensals. Okay, does anybody know if Anna wants a sample of these corals? If anybody's sitting with her, perhaps they can ask her. Maritza asked for one. Okay, so there, Anna said yes. So I'd like to sample. I'm assuming we're not going to take the whole thing, but just snip the top.
Yes, I want to.
This piece.
Thanks.
Yeah, off the top. Okay.
Can you put like big salt in it? Yeah, I already. Okay.
Does she need another Niskin? Somebody asked. Well, she doesn't have a Niskin yet, so.
She did say
Okay. We'll just confirm. Now that we have a lot of sediment, we don't want to trip a Niskin until it goes away. So that's number 2 you just opened?
Yes.
So after we snip the coral and the sediment clears, we'll want a Niskin sample here.
Okay. You can return it.
Photo of what?
Yeah.
The
I
crabs?
just can't see, so.
Yeah. Give it a little thought. Turn a little forward.
I wonder what phase of the tide we're at. If we're in slack. And why don't you guys look up the tidal cycle for here and just see when, whether we're in a low or high tide or not. Oh, well, we'll take it. Guys. Erin, go ahead and record the sample if you can, please.
Sorry about that.
So it looks like we have some ophiuroids along with it. Quite a few. And the polyps are all retracted. Their tentacles. Okay. Will it fit in the quiver? Okay.
Hope so.
We were trying to snip off the top, but it doesn't always work.
Come around a little bit.
USB or okay. Thank you, that looks good. So we have a request to photograph, image the crabs.
Okay.
Do you see crabs on the wall there?
Can you tip this back?
Let me see.
Yeah.
Okay,
Oh.
thank you. Did see.
Yeah, can you tip this full little one?
I'm trying to get it lined or?
Yeah.
Fold it over.
Yeah. Okay. You said you wanted the fire in Niskin, yeah?
Yes, yeah. So it would be Niskin number 4.
Yeah.
One failed. We fired 2 and 3 earlier.
Yeah, you're correct then.
It doesn't work like that, you gotta hold it.
It was fired, but we were told it's actually fired even though visually looks like it wasn't. I mean, maybe the pilots can confirm that for us, but
There you go.
Okay.
4.
it's number 4.
Unfortunately, 3 and 4 are hidden behind. C3 is definitely on. 1 didn't fire. You have 2, 3 definitely did. 4 is behind this pillar, so it's really hard to tell. Other than the indication of the flag. And 5 and 6 you can tell.
Maybe you could look at the P4 on your right. Let's try to put the camera on.
So go over there.
No, no, if you put it way in front, you might see it down the way. Then put the flags on.
I think we're okay, as long as four, you can tell four fired, right?
You think so?
We think so, but we can't see the canister, I see, the Niskin. Okay, so Pachi wanted us to zoom in on the crabs, is that right? Over the corals. Okay, let's just do some high resolution imaging over the patches that we've been sampling. Okay, so you want the crab that's on the coral?
Yeah.
Where'd he go? He was there. Maybe he saw that one other coral get taken and decided to take off. Or maybe he's behind. Don't see him anymore. Might have walked off. Ooh, there's a sabellid polychaete right there. Beautiful, there's a couple of them. Fan worms might be the common name. There's a brachiopod behind it, and all sorts of draped fecal casts coming off the sediment right here. And somebody underneath there. Don't know what that is. That's a crustacean. There's another small anemone. All kinds of things underneath. So one question is whether the heteropolypus, the bigger coral-like structures, create a habitat under that protects animals. And we're looking, it's like in the desert, the big cactus provide a nursery habitat underneath them. It's a little different and moister, and here maybe there's food accumulation or protection. There's also a couple of very small polyps sprouting off this tube. Okay, can we scan over and look a little more under a few others? I'm just really curious if there's aggregations of animals under these. Or not. There's a nice view of the lophophore also in this one.
There's a crab on your right.
This one has sponges growing, or maybe other bryozoans growing on it. These are probably encrusting bryozoans. And this is on the brachiopod. This might be some sponge.
Salt crabs. Yeah, just on
Let's
there.
pan over, see if we see crabs. There's some serpulid polychaetes, are the little white S.
So I missed
Right here.
only one on the bottom left. It seems like behind, right there.
Right back
Yeah,
there.
This is like. I guess I think that's the.
Crab is hiding. We have lots of arborescent foraminifera. That's what these little tree-like structures are. The little white calcareous tubes shaped like an S or a C here are serpulid polychaetes. There's the crab leg peeking out.
There's another one there. See that one?
There's a closed-up coral. The common name is mushroom coral. You can see that when they close, they look a little bit like mushrooms in the ground. Okay, we're being asked for the sabellid that we saw earlier. Let's see if we can find a way to collect that. That was the fan worm that was in that first coral we were looking at. Elohi has asked us to try to collect that. It was
Straight.
maybe right there. I don't know how would we get that without disrupting the entire set of organisms. I don't know how to collect it. Maybe, I wonder if we could find another one. Let's just ask. There's one. God, it keeps changing. My phone keeps changing Sabellid to Danielle. I hate that. So that's the only ones we've seen, huh? Well, yeah. You can try with the manipulator. I think it'll just pull everybody off there, but I don't know if these guys can reattach the big corals once they're pulled out. They'll become food for shrimp or something. Oh, it just went in to its tube. Thing is, I don't know how far back in that tube goes into the rock. So unless we I think we should look for another one, because I'm just thinking that there's not going to be any easy way to get that without just sending. You're looking at this right here. Yeah, I don't know, other than pushing the whole hillside, going behind this whole clump, because they could be long. And then we could start an avalanche, sort of. That's what I'm concerned with. So why don't we keep, I'm sorry, what did you say? Oh, the snail. Yeah, the snail's probably of interest too to people. Maybe other little anemones, infaunal anemones that live in the bottom. Then we have lots of different kinds of fecal pellets, long ones, sausage-shaped ones, and pellet ones. Oh, there. That's maybe the tail end of, well, it might be feeding. Some worms feed that way. It's not a snake, but it's polychaete. Interesting. I love the spaghettis. These are spionid polychaetes often. All these piles of mud spaghetti. The oval ones are made by capitellid polychaetes often. There's a couple groups that are subsurface deposit feeding worms that always extrude their pellets onto the surface. All right, I just don't think we can get those sabellids without totally disrupting everything. So there may be others. Ooh, these white tubes are interesting. Could we zoom in on them? I think these are a protozoan called Bathysiphon filiformis. I might be wrong. This protozoan single-celled animal makes these long tubes that look just like polychaete tubes. And I don't know if these are polychaete tubes, but Bathysiphon filiformis looks just like this, and they occur in somewhat sloped, organic-rich areas. Okay, Maritza asked for the small snail. So maybe we will go back to where those tubes were. We haven't moved, right? We're just panning around. Maybe we can section some of that. Is that the spot?
Yeah.
All right. Can we section up that snail, probably? And there are a couple of these tubes here. I see, there's a small one there. So maybe we would get the sabellid too. has got a shot of the organisms, the snail and everything. The brachiopod's open. This is number 4.
Yeah.
This is number 4.
Yeah.
You may have got one of the sabellid tubes too, I'm not sure. Well, there's still a couple, they're still here. I guess these were the small ones, but
It's not there.
What's up, buddy?
It's not trend.
but move along.
Yeah, sometimes.
We're looking to see if the snail has entered the suction compartment. Sometimes it takes a while. It's quite a long tube.
Take it up.
This is one of the finer mesh
Is it?
sections, so that we might end up keeping some of the mud, but it's not a big deal. But we don't want to take up too much mud.
Right. Okay.
Hello, Ben.
Good morning, Lisa.
No.
Confirm.
I think we got a snail net, we just didn't see it drop in the tube because they were dirt with it, and we can't confirm. Dirt was a little way to the hillside anyway.
It's interesting, these these heteropolypus or whatever they are, when they pull in, they don't go all the way to mushroom shape. They just sort of end up with sausage arms. So maybe there's something different than the Anthomastus. Some of them pull all the way in like that one. But so was the verdict is that we may or may not have a snail?
Yeah.
Keeping the delta around 60, we're going uphill. So past the vessel is where you start moving, so you can have to keep the water, so we're not running our uphill against the hillside. Just working on the uphill.
This is a different type of coral garden
I know you're gonna sit down.
than we're used to seeing, but really interesting. Dominated by these heteropolypus cnidarians.
Niskin 1 didn't fire in. It's open, so we did 2, 3, and 4. But we can confirm it 4. No, we did see 2 and 3. But 4 flagged and went, but we can't see the tie wrap hanging down. So we think it's fired.
We're changing the watch. I'm gonna say goodbye to everybody, and we'll have Greg Rouse join us.
When we're doing deck checks, the port seal bearing compliant.
Thank you, folks. Thank you, Doug.
So I tried tightening it and it started leaking more. So the port bearing compliant.
We are making our way to waypoint 2.
No,
So we haven't begun any of these
we
100
tried to
meter
tighten it, it
transects.
started
We
leaking
did
more.
a 100-meter one in the end.
Swage ferrule and so when we took it out,
You
the
don't have
swage ferrule.
just cnidarians. But it'd be good to do.
So we made a plastic one temporarily, just in case we get to die.
But
Maybe,
you can't
the
stop
two
the sample
we
or
used to
zoom.
compliant and get in it. I made that one with that. Put that in
We've
place.
been
And got a piece of stainless steel and with some pipe and we bent one in shape. It's
No,
not
we
exact.
aren't at 2 yet. We were making our way to 2, but not doing a transect, so I said to stop and look here.
So it's good. And the plastic one, the gathered on the next part of the
Got everything.
Did we get those Sabellans in?
No, we would have had to start an avalanche. We got the snail that was next to the Sabellids, and I thought maybe the Sabellids would come. But it didn't. So if you see more of them, you can try to get them.
Well, we'll do a transfer.
They're tucked really deep into under the coral and
Yeah,
walls.
and then
So,
choose.
But I think that's kind of.
they do.
I think what we should do is, when we go on there, change that.
You can obviously,
No, change that, in a scan, this one, would you, thing?
I took one yesterday, so I assume that one was for Calhoun's, because we put 2 originally, so.
One with the we could try it out and see if it's out.
A new one.
How dogs are. I want to do that. Right. Let's turn you up.
Okay, I didn't get the one. The really, really long arm one.
That's sitting on the red one.
I do.
Red.
I do.
If I can't find
Top of the morning.
Okay.
Feature here. Once you get to waypoint 2, looks like it. Left is off. This is the good part here.
Where's the good part? Where is it?
We're in it. This is
the steep part.
The steep part always seems to be the best part.
Now it's going to get boring.
Look at the penetration. I don't know what the exaggeration is, but
I hope not.
2.
2. That's pretty good.
Pretty steep.
Yeah, this is some juice here.
What's the plan, Disney?
We will head to waypoint 2. Thanks. And then I think we might knock out some of these transects.
Okay. Ready? Shall we?
Yes, please.
Port ram is deployed and anchoring near the bottom there.
Afternoon, everybody, from off the coast of Chile. We've moved a little south from yesterday. We're in a place called the San Antonio Canyon. It's still pretty much offshore from El Quisco. And as you can see, we're at about 1,000 meters. We're doing coming up a canyon wall. From shallow to deep is the best way for the ROV to operate. And earlier we were in the bottom, the axis of the canyon, where they did some mud coring, and now we're coming up the slope, which should be fun. We've got some transects to run and to document the biodiversity, and we'll no doubt sample a few creatures along the way. Lots of squat lobsters, lots of an octocoral, colonial octocoral that we're not quite sure of the name. There's multiple possibles until the taxonomist looks at it. Anthomastus, heteropolypus. There's squat lobsters and strikingly lots of white brachiopods, little mollusk-looking shells, but actually a completely different group. And I'm here with you today with Anna Damo, and Anna is a coral expert, and she'll be helping us choose which of these octocorals we'll sample along the way, and maybe we'll get lucky and see a true... Are you interested in stony corals? If we see them? Yes. So far, no luck.
No.
And we've got Fernanda and Peter, who are helping us with the logging. And Ben and Levi are our pilots today, for the moment, for 2 hours. So brachiopods, those white shells you can see there, an ancient group. Ah, there's a clam. Oh no, it's an anemone. What do you think?
Zoom in. Go ahead and zoom in.
Could we zoom on it? Thanks. Oh, that's a clam. That's a cesta. We could grab that one.
before we start doing transects, please?
Where's it again? Can you point on it?
Oh, sorry. I think it's there. There's one there.
The fuzzy come out of it?
Yeah, the little orange tentacles.
Yeah.
So that's a real bivalve, not a brachiopod. Acesta have very delicate shells, common on continental canyon slopes like this. So we'll try and get one here.
Which is still that one, you think? Box or whatever.
Oh,
How thin a cover?
I think this might be a slope.
Slope?
Yeah, it's going to be pretty fragile, but
This one?
this one. Yeah. Think that'll fit?
Is there anything hard about it? Is it a hard shell?
It does have a hard shell, but it's very, very fragile.
You think you can
No.
Too big?
Not too big for a cover, right?
No, for a slope.
It's too big for a slope.
Okay.
Let me see.
Maybe the scoop could just
Ledges are out there. It's
Yeah.
like 5,
5 across.
6 centimeters across.
It's gonna get jammed up. Put it in a box.
Yeah, with a scoop. I think if you picked it with the manipulator, it might break. But we could try, because I'm sure that we'll see
The
others.
jaws might crush it.
Yeah. It'll be attached there. Just
Whatever you feel comfortable. You wanna use the basket, or you wanna pick it up? I would pick that up.
Okay, yeah, we can try picking it up.
what would you try, Ben?
It says use the
Yeah,
rubber.
try the manipulator, and I think we'll see others.
Just gradually close it. I need the steps. I guess we'll put it in a box. Box.
Yes, thanks.
Let me get it.
So you think it's too big for a quiver? Yeah. So
So
this acesta, with that fringe of tentacles around it, is related to swimming clams like lemur or file shells that can swim, but this one is anchored. It's ACESTA if you want to look it up.
Got a good sit picture too. A little bit more. See if we can go like a level arm or pointing out in the forearm. There you go. See that? There you go. That's it. That's all you see. Okay. Now you open your jaw a bit. Stand by. Okay.
There's been studies on this genus. They seem to stratify by depth. So this could be Acesta sphoni or Acesta mori. We'll do a sequence and check. Or it might be a different one altogether from the ones further north off the west coast of the US.
Okay, no. Close it. Just right here.
Very well done. Oh, and it's...
Stay on there. It's still down there. I see.
Took a tumble. Well,
Between those two red circles.
Yeah, I think it's here.
You just want to try and scoop it. So, feel happy.
I think I should put jaws quite
Yeah. After you grab it, don't hold it right away, just to make sure. You're in the middle. You need some soup. Hold on. One more. That's, scoop it underneath, right? No, one more there. That's it. Beautiful. Easy.
Good job.
Let's see. Hold on. Can you turn it? Oh yeah, it's intact.
It's good.
Okay.
We can definitely do it for one valve, I think is good.
Move on.
Anna, talk to our Spanish listeners about the plan today. Talk in Spanish about our plan today. The transects.
Hola a todos. Hoy hemos empezado a mostrar un nuevo sitio de San Antonio Canyon. El plan es un poco distinto, de mostrar un poco distinto de lo que hemos hecho ahora. Hasta ahora. Estamos explorando la pared del cañón de San Antonio. Y vamos a seguir un transecto de muestreo que va desde la pared del cañón hasta el centro del cañón. El cañón es muy largo y muy ancho, por eso por el momento estamos explorando una parte que es más, digamos, cercana a la parte más, digamos, de la cabeza del cañón, de la parte de arriba, del principio, cerca de la costa, y luego vamos a ver en los próximos días otros planes. Y en este momento estamos recolectando una almeja. Ha sido bastante difícil, pero el desafío para los pilotos del Aerovisen es bastante grande, pero les gusta, así que han conseguido efectivamente mostrar esta almeja que tiene las bivalvas que son muy frágiles, entonces no es bastante delicado. Y una vez que hemos acabado de recolectar esta muestra, vamos a, como he dicho antes, vamos a ver hacia el centro del centro del cañón, perdón, y a ver de mostrar probablemente
Great. Well collected.
otros organismos que están en el centro, para ver si es la misma comunidad entre la pared y el centro del cañón.
Okay. Now we better get to waypoint 2, or we'll be in trouble. We'd like to do 4 transects in the next couple of hours, which means we will fly above the bottom without stopping or zooming. Just looking with the lasers on so people can map what we see.
Onward and upward.
Yes, please.
Alright. I definitely squeezed it a little bit, but I hope I didn't crack it.
I don't think only one of the valves cracked, so it's
Okay.
perfect for taxonomy. No, well done.
Esta pared es llena hemos visto desde esta mañana que la pared es llena de octocorales,
que todavía no se sabe muy bien no sabemos muy bien qué tipo de autograde de género. Podrían ser varias oportunidades, varias posibilidades, así que hemos recolectado una muestra para unos expertos chilenos que luego vendrán a utilizarla para identificar. What is that one? Greg. What is that?
Can you just look down, please, Ben? So-
Yeah, that one. Maybe it's just
This one?
Is that
It's a
sponge?
It's a sponge.
Who's that? The shape is
Okay,
looking
we can
very
get that.
nice.
You want to sample that?
No, thanks.
Okay, I'll keep it.
We better get shallower.
Alright.
We've been here a long time. It's the trick if you've got to do these transects is moving along. But this morning's watch, they had to do a lot of push coring. It was tricky, so that stopped the transects really happening.
June was like 17 or 16 yesterday. What does it feel like now?
It's heavier.
Okay.
So a very steep part of the canyon wall now.
Greg, we have the inset of imagery there for reference of how steep this face really is.
Oh, look at that. Can the audience see that?
Yes, just
Yes.
a small inset there.
Okay, yeah. So John's shared an image of you for you where the little yellow dot, the white line, is our track. And the path we're going to follow is the zigzag. So we're trying to get up to the next white dot, and then we'll do a transect across. And then we might do some more sampling. And then we'll keep alternating with sampling and transects.
This is a good distance for you, Greg, when you want to get closer.
I think it's pretty good, Ben.
Yeah, so
Yeah.
I was thinking we could see more.
Yeah. Still mostly the octocoral, galatheids. We're seeing still a lot of the brachiopods. Some sponges are showing up now. Some brittle stars. There's an Enypniastes. We collected one of these earlier. Benthopelagic sea cucumber. They can swim along, but also settle on the bottom to actually feed.
Did the first watch do a transect? Do we know if there's been any transects?
I don't know.
Notes about transects?
I don't think so. Ah, yes. Transect. 13, 20. Two transects. At 10, 9.
Yeah. Where did they do the transect? 2, waypoint 1.
Here is not written. Translate start and end, but they didn't say where.
Do you have anything here? On the last side?
Large anemone in the bottom of the field there. Same colour as the octocoral. Little octopus. So we'll just hold it, waypoint 2 then, and then we'll start a transector 3.
Yeah, this one, I keep turning because I'm going towards the turn. This way or something.
So, loggers at waypoint 2, we're going to start a transect. So between 2 and 3. So if you could fill it out, just note on the sheets, the dot. And I think in the sea log, just making a note of start
end.
transect. When we start, it'll be, we'll call the transect WP2 into WP3. Thanks.
Greg,
Yeah.
here, which comment do you want? Because waypoint 2 and this is waypoint 3.
Yeah, we can ensure it on this side.
And do you want some special comment? Specific one? Like because start and end is here. The start and end is here. But here, any other comment that you want to? Okay, what we see.
Some large yellow sponge there hanging on the wall.
Oh, that nice. The color is amazing.
Transect will go to waypoint 3, is that correct?
Yeah, thanks. And we won't stop, we won't zoom, we'll just follow at the distance Ben had kind of set was pretty good.
So that's 100 meters bearing 1, 3.
Are we on 2 now?
We're almost.
Just about? Okay.
Cool wall.
Yeah, wow.
You want to stop it?
No, we'll keep going.
We are on
Okay. So let's start the transect. And it's a little more horizontal rather than
Yeah.
vertical.
Yeah. Over here. Moving away from us.
Your waypoint, thank you.
Gregory?
It's been
It's a traveling transit beside the wall. So I'm gonna pan the camera to the right, and then I'm just gonna fly my, I'm gonna be looking at a weird angle.
I see.
Decide to go fly this side.
Yeah, when it's, should we rethink how we set these zigzags with your, should talk
No,
to you?
I'm okay with
It's okay?
it. Just to let you know, so when you're rigid, when you're looking at it from weird angles.
But I mean, the goal is to be able to count and look at things. Should still be fine. I mean, the goal is the image. Yeah. Yeah. Is it better if we just vertically transect for the next ones? Or?
No, it's good. It's okay. Just a little bit further out.
You said when you're starting. We'll log it. Okay. Huh? Okay, we're starting now.
¿Está al lado?
Do you want to kind of send the horizontal? And then start climbing up slowly.
If you can just follow the line to 3, that's all we need.
Same depth.
Yeah, off the bottom. So from here we'll be here a few dives, and then we'll head south to Conception and look at Seeps for a few days, and then also look at the Conception Canyon. And then on the following leg there'll be more canyon exploration heading south to Patagonia. And the idea is to be able to compare these transects with the ones they do subsequently and document the diversity. So we'll mostly choose the canyons to be at similar depths. we started this particular transect at 986 meters, and we're going across the canyon wall a bit, so it won't be so much vertical gain. But you can see the map of our path is this nice octopus there. Our path is zigzagging backwards and forwards. That actually makes it challenging for the pilot to fly and keep a steady distance off the bottom.
Can I speak in Spanish? Lo que estamos haciendo ahora es, hemos empezado el transecto del punto 2 al punto 3 que podéis ver en la pantalla, y la idea en este momento es hacer solo una visión de lo que hay, y vamos de punto 2 al punto 3, vamos zigzagando y intentando hacerlo. Y como podéis ver, hemos subido por la pared. Estábamos en el punto 1 antes, que era como por debajo de la pared, de la pared del cañón, y ahora estamos subiendo hacia el punto 3. La idea es solo hacer, como he dicho, una visión de lo que hay en general, y durante el transecto no podemos parar porque no vamos a mostrar nada, solo verlo y anotar lo que vamos viendo a lo largo del transecto.
We're starting to see some crinoids now.
Oh, yeah.
We've come up only 20 meters from where we started this transect. So vertically 20, across 100. Just about. That's a rat-tail macrurid and another crinoid. Pretty steep on this section of the slope. Okay, end of transect. And we came up to 955 meters. Thanks, Ben. Well, why don't we just continue over to 4, because it's at the same depth. So we'll start a new transect to 4, and then we'll stop there and have a look around. Thank you. We mapped this canyon last night, or the ship mapped it, and our colleagues Adam and Jeff looked at the data. We were trying to see if there's any seepage, which would have altered our target. But nothing apparent, so we've just stuck to our original plan of looking at the walls of the canyon at about 1,000 meters, and tonight we'll map again out at about 2,000 meters and then dive tomorrow at that depth. So it should be quite different to what we've seen today out there. I'm hoping for some sea peaks. That's a Brisingid sea star.
Mm.
They stick with their arms up in the water and catch plankton. A little reminiscent of the basket stars, but they're a sea star, not a brittle star.
Yes. Lateral. Yeah, lateral north. Northwest. One north there? Yeah, it's.
Hey, Bridge, it's RV. Who do you got?
Yeah, wait.
Keeping your delta at 60.
Two, three Anypniastes in the frame there. When they feed, they fill up with sediment, and that'll keep them on the bottom. But if they void that out, they basically become neutrally buoyant, can easily lift off. Now there's another sea cucumber.
Oh.
That's a deep sea form. Nice to see. Hopefully we'll be able to catch one of those. It's quite a large one. Looks a little like one called Cycropodes, but that has a tail. So it's probably another one, Lake Gamoni. We haven't seen a lot of rock on the last more than 150 meters, so hopefully we'll get a nice escarpment or something. The rocks will give us a lot more fauna to look at. Good. Okay, that's the end of transect 4, we'll call it. No, transect 3. Transect 3?
Mm-hmm.
And we came up to 923 meters. Anything hard on the sonar? Well, let's head to 5, but we won't transect. We'll just look, maybe collect. I guess there's no reason for us to go to 5 if we're going to end up doing another transect at 6. If you see any hard target, we don't have to stick on this track. So the way this is set up, if If we did 4 to 6, it's 100 meters. If we did 4 to 5, it's 100 meters. Or they're all, every line is 100.
It's different times.
The bridge, we got a new waypoint.
New waypoint.
This would be flat, right?
Yeah. Maybe at 5 we'll not go to 6 and we'll just go 7. See if we can get some elevation.
Okay.
Let's head to 7. Forget 6. We won't transect it. We'll just go there. Try and get up a bit high, shallower. See if we can collect something along the way. We've got 25 waypoints, so it's a long way to go.
Tell them I made an adjustment.
Although we have
The bridge, we got an updated waypoint.
we've got 6 more hours scheduled on this
Okay, thank
path,
you.
so we might end up at 600 meters. Could be doable, or 650. So if you can see our little map, we're going from waypoint 5 to 7 now. We're skipping 6. Got lots of large shrimp on the bottom there. They're heterocarpus. We saw them at the Seeps of Elquisco the last few days. Notice how they're all pointing in the same direction. basically upslope. Maybe something over the current. We're still looking for a rocky outcrop if we can to do some sampling. But nothing showing up on the sonar yet. Tonight in this canyon we'll do a net tow for Fernanda, who's a student here on the cruise, and she's after larval fish, so she'll be dropping the net over the side, and we'll do a CTD, which is an instrument to collect water, but also measure things like temperature, oxygen, salinity, as well as depth. So even though we'll be processing the samples from the submersible, tonight there'll be other people actively deploying instruments over the side. Still no hard targets coming up. I don't want to collect an octopus. Let Olafio collect it. So we're nearly at 900 meters. We haven't seen some rocks for quite some time.
Go ahead and push it. Yeah.
I think we'll keep going to 9. Thanks, Ben.
How does that look, man? That's good. The bridge, got any waypoint for us? It's out there.
Yeah, wait. Can you zoom out the picture? Or just trace?
Pretty much normal to
So it's really kind of, the slope is less steep. Maybe that's why it's so sedimented. No, thanks. That's what we were seeing a lot deeper, but it's the first one we've seen for a while. You can see from the map we were initially pretty steep, so a lot of the sediment would slump off, exposing the rock underneath, and then animals could hang on to it. But now we're on a bit of a shallow part of the canyon wall, so we're just seeing a lot of sediment. So the canyon's really dominated here with the heterocarpus shrimp. There's quite a lot of them macrurid, rat tails, and then Enypniastes in terms of the visible animals. Okay, we're nearly at waypoint 9 now.
Still there. Shoot that up. You should fill up, right?
I think so, yeah. Let's just do 11.
Extend the ship after that. Bridges, are we got a new one?
Yeah, wait, wait.
Since we're not stopping, I'm actually writing these down as transects.
Hmm?
Since we're not stopping, we have just been doing transects. So
I recorded. I didn't record it as a transect.
it's okay, I did.
I recorded here, but not because you say stop and you didn't say
I
start.
didn't say anything. I thought we were going to stop, but then we didn't. So I think we can retrospectively make them.
Yeah. And so will be like unique transit, transit 3,
Yeah, so
finish now.
2 to 3, 3 to 4, 5 to 7, 7 to 9.
So transit 6 now. Yeah. Just give me.
I even made this 2. This was partial, so we can make this 5.
5. Give me just a second.
Sorry, everyone, we're trying to
Transects.
figure this out.
Okay.
Again, we're still on a pretty flat part of the canyon. I think that sea cucumber's too big.
What do you want to play?
Yeah, we won't take it. It's 1/2 meter long.
It's pretty barren.
Yeah. Nothing on the sonar.
Sea cucumbers before. You say for sea cucumbers. Oh. That's why it's stupid.
Yeah.
Just gotta read it. And then kind of still put the jaws on it.
If we saw a smaller one of that purple one, we could go for it.
There's a bigger one.
Well, let's have a look at it. Let's get some nice video for a bit of a change. So here you see a wonderful sea cucumber, probably a Cycropodidae is the group. So a good 30 centimeters to 40 centimeters long. Often they have symbiotic animals associated with them, shrimps or worms. Can't see any on this one. Just try and get a name on it. There was quite a lot of activity off the Chilean coast 150 years ago with the Challenger expedition, and quite a lot of sea cucumbers were named off this coast. So this might well be a known species. Challenger trawled all the way down to the bottom of the Mariana Trench on its journey. It was a steam-powered sail ship, so it had a sails, but also a steam motor so that it could actually do trawling. And they had hemp rope to get down to the bottom of the Mariana Trench. Quite an amazing feat. off Chile, they were routinely trawling down to 4,000 meters. Okay, I think that's a nice view. Maritza suggests that it's benthodites. And yeah, that looks like a pretty good identification. Thanks, Maritza.
Do you know what that scene in the middle of it is?
Damage. Yeah, or it's contracted, but yeah, it's not normal. We could keep going. Thanks, man. Yeah, I don't know, Peter, what would cause that. If we see another one, I doubt that it would have that same mark. You can look up online, bentho, B-E-N-T-H-O, dites, D-Y-T-E-S, all one word. It was about 30 centimeters long, so they get much bigger. Even that's difficult for us to manage though with our collection jars. So we'll be looking for a small one if we want to collect. There's another one coming in now. This one's even bigger. So we're just about waypoint 11. Very flat here. So we'll just continue on to 13. Thanks, Ben. Thinking, looking at the map, things will get better at 700 to 600 meters. We're away from that, so we might want to run through some of these transects. Oh, you got a target ahead? Look at that. Okay, we might have something interesting coming up. Just a depression, not a rock. Great image of an Enypniastes there.
Smaller ones are flying. The big ones are like that.
That's the back side of another one. Well, there's a smaller. This is another elasipodid sea cucumber, or it could be a synallactid. We'll see. This one's big enough to collect, or small enough to collect more accurately. Some sea cucumbers have pretty strong body walls. But others are very delicate, and once the pressure comes off them, they just seem to liquefy. Yeah, you can see the sediment. This one's dropped. Think we could slope this one? You pick it up? Okay. Let's get a nice close-up of it. Again, this one doesn't seem to have any symbionts associated with it. I'm always on the lookout for little shrimps or worms living with these. Maybe pull back a little bit. Yeah, great. So a quiver or a firebox, I think. Firebox is easier. Could go in number one, even with the fire.
You got video?
Yeah, we're good. Thank you.
Can you give me a instant picture? The one on the right. Yeah, sorry.
No.
Huh. Whoops.
We felt that he slipped.
Yeah. Lucky day. Hopefully it'll get off the basket. Otherwise. Yeah. Okay. No worries.
I find it almost as easy just to catch them on the fly. You know, about one. Like when they're flying, porch out, and then I open the basket and then swoop them. We've done that before, swoop them, fly underneath. Okay, you wanna try that?
Oh, that's an Ipniastes. We don't need that one. We got one earlier today. So we'll look for more on the bottom. So this kind's more squishy, and it's got that big flappy part at the front. The other one you saw wouldn't take off. If it had been in an Ipniastes, it would have tried to fly away.
It's close. This is one of them.
Yeah, they're not far apart. They're fairly closely related. Yeah. Keep going to 13.
See what I'm talking about? When they're flying in mid-water, it's easy enough to watch out, open the pipe up that's underneath it. This way we can
Right. Oh,
put it in the
yeah.
box.
Okay.
That's how I would catch the squid, the giant squid.
Squid let you do that?
Yeah.
Where's your RP? We're gonna update with.
Since they don't get spooked.
So if you're watching earlier, we did a lot of push calls. Actually did 14, 13. So that was on the last watch with Lisa. So we don't have to do any more. So that's good for us. We can just explore and do transects. But we'd really like some hard substrate to get some of the other creatures that live here. We started this dive about 6.5 hours ago. We're coming up on 13, number 13 waypoint out of 25 that are marked. So we're about in the right position if we want to get up to 600 and...
Not this big one?
No, we got nothing to put it in. We'll take a baby one if we see it. Skate to the left. Skate to the right.
Updated waypoint bridge.
Update.
I guess if we get here, we'll get a bit more interesting. So let's start a new transect when Ben calls this finished. So that'll be from 13 to 15.
We're here. 13.
Thanks, Ben.
Headed to 15.
We're on a transect now, so we won't stop. That Benthodaites is nearly 50 centimeters long. Sea cucumber's closest relative is sea urchins. If you think of a sea urchin and basically stretched it out and took away its hard test, you'd have a sea cucumber. They still have a skeleton, but it's reduced to little calcareous rods and wheels in the body wall to give them some support, but otherwise they're largely very soft. They diverge from sea urchins. The sea cucumber branch and the sea cucumber and the sea urchin branch split from each other about 400 million years ago. Ah, there's a nice urchin that's like one we saw in Limari Canyon a few days ago. Every now and then there's a big burrow. I've always mystified us what lives in those. They're very hard to figure out sampling those burrows. Probably some kind of trap for getting whatever it is. Probably crustacean. But there are other things that live in burrows that wouldn't come for a trap. Various giant worms that no one's ever been able to sample. Coming up on another big Benthodaites. Look at the lasers, 10 centimeters, and estimate the size. It's at least 30 centimeters. Notice how all the shrimp are facing in the same direction. We're still on a pretty flat part of the slope, and the Benthodaites can maintain their position. If they'd been lower down, there's no way they could stay where they are on the slope, and they would tumble. So here's a much better place for them to live.
The red one's flying, right? Is the same species that you wanted?
No, different. Yeah, we collected one at the beginning of this one. Yeah. So that one's nickname is the headless chickenfish. It's not very nice. I think they're pretty cute. There's a nice big sea star coming up, sea urchin rather.
Sea urchin.
Yeah, so Nico, who is no, we'll finish the transect. If we see another one, we might grab it. But he tells us that's probably hygrosoma. We saw some before and thought they might be tromicosoma, but Nico thinks it's hygrosoma, Hygrosoma. Another Benthodaites there. And we're coming to the end of this transect. We've done about 100 meters, but we've only come up 10. So we've been on a pretty flat plane.
Passing 13, 15. Going to 17.
Okay, so transect is finished at 16:40, and we're heading to 17. We can just continue with the transect numbers. So let's start a new transect.
Bridge, we got it.
And once we get to
Yeah,
17,
wait, thank you.
17, we might start to get a bit of a slope. To see an enemy, first one we've seen like that. Yeah, hopefully. Little skate there in front of the Amphibites. Very small little skate, just 10 centimeters, plus the tail. Again, this transect, we've barely come up 8 meters over the 100.
17.
All right, thanks, Ben. And let's head to 18, and hopefully we'll get a bit of a slope on this one. Sorry, not 18, 19.
19.
Heading to 19. So we'll do another transect to 19.
Roger that. Okay, turning left.
Oh, we're,
We're on 17.
yeah, so we're 17. We're going up here. Is that 19?
Oh, my eyes can't see that.
Yeah.
So look at my screen here. See over here? We're here. 18 would be directly uphill.
Yeah.
Oh, but oh, that's uphill. What's over here? What number is that?
17.
No, it's 21.
That's 21.
Let's see what's on the map.
It's 21.
Yeah.
Okay. Is that only 100 meters away?
So from us to 21 is 150.
150. Okay, no, that's not.
We can just hop up to 18 here.
Yeah, okay. We're on 18. Oh, sorry, we're going to 18. If we do 18, from 18 to 21 is 100?
From 18 to 21, yeah, 120.
Okay.
You know, it was 18.
I want to go to 21.
Okay, you just head straight.
Let's go to 21. We won't do a transit.
Roger, 21.
Yeah, let's just look at 21, get some elevation, and hopefully some sand. We've done a whole lot of transects now, so we've got lots of data. We'll try and get a bit shallower and a little more steep coming up. Hopefully there'll be some rock. Yeah.
It's a good speed for you.
Yeah, that's good. Thanks, man.
We are 18, heading to 21.
Wow. There's another one of the benthidites with a big squish in the middle. As if a manipulator squeezed it.
There's side tabs on terrain here. Let's go. Let's see if it's the maybe you want to take a look?
Yeah. Yeah, we're not on a transect now. We're going from 18 to 21.
Yeah, but you say start a new transit, so
I did.
and then you stop. Okay, no problem. I can just.
No, because the distance isn't 100.
Okay.
Oh, wait, we're not on the transit?
We're not on a transit. Well, we stopped at 18.
Yeah, 18 I got it. Excuse me.
It's just a small.
Yeah, let's have a look at this. There's a lot of brachiopods. Yeah, it's a little octopus. They're called brachiopods, I think. We'll have a close-up. We can have a look at the octopus. Everyone likes to look at the octopus.
Greg, what kind of birds are we looking at right now?
The shells are brachiopods, or their nickname is lamp shells. And to look up that, they're an old-fashioned kind of lamp. And we've of course got the octopus there, macrurid, rat tail, and the squat lobsters, and the little octocorals, the pink polyps, and even an anemone. And you can see the brachiopods look like mollusks, like bivalves, but the ones that are a little bit open, you can see a curled structure. Although right now we're interested in the octopus, so we'll look at a brachiopod in a minute.
Uy, I saw coral.
Amazing skin they have. Ah, hello here. I don't like collecting octopus.
No. No, Ben.
Nope, nope, nope. Too big. See, look how big it is. It's enormous. Can't catch that. Can we have a zoom on the brachiopod? Let's see.
Hey, coral.
Oh, and
We saw a coral.
wait.
I see.
Anna, do you want to point out the coral?
There, no. Zumba.
Okay, someone's excited.
Cute.
Okay. Okay. Great.
But just to check, no?
It's a solitary coral. Yeah.
Yes, just to.
We could sample it. So it's just this one. Is it Desmophyllum?
No,
No.
because the septa are very regular.
Okay.
Yeah,
It's a little
could
cup
be.
coral.
Yeah, could be Caryophilia or
Caryophyllid. Okay.
oh Yamania. Yeah, looks like more Caryophilia, but it's so nice. Finally, it's so well.
Okay. So as you can tell, Anna studies corals, and we'll try and collect this. You can see the brachiopod below. It's open, and it has a structure called a lophophore. Inside, it's a filter feeding structure. We're going to switch out the pilots now. Thank you, Ben. Hi, Doug.
That crab is. For what?
Coral.
No, just one.
You don't want it?
No, leave,
Leave
it.
it?
Yes, just one, leave it.
Anna
Maybe
wants to
can
leave the coral.
populate.
We'll leave the coral. Doug, I'm just trying to find a nice view of this brachiopods where it's open and you see kind of a horseshoe shape inside.
Ah, that one.
That's all the zoom I have.
Yeah. Oh yeah, there we go. That's good. This shape's good. That's great. If you can center up that. Perfect. Okay, and that might help us identify that later on. So it's a little tentacles covered with cilia, filter feeding. If you want to look them up, they're called brachiopods or lamp shells. Okay, thanks, Doug. We can keep having a look around.
Do you want to stay stationary and look around, or you want to?
Stationary. Yeah, we're still seeing if we want to sample anything here. We could get some more of the brachiopods actually, and that's a sea squirt there and there. So the thing you can see that's white with a hole at each end is a tunicate. And there's a gastropod up there, so we'll probably we might slurp this area to get that, and maybe knock some of these shells off. We've got a great view of the lophophore of the brachiopod there in the middle. Oh yeah, and a sponge on top of this. Yep. Yeah, nice sponge sitting there.
Yeah.
I can see it. Yeah.
Can't tell what it is.
Yeah, it's beautiful. And the little, this yellow filigree, that's its gonad.
Oh, okay.
Yeah, eggs or sperm. Oh, they're just closed up for us.
See it real good.
Out of here.
Yeah. These brachiopods have a little stalk that they're attached to the rock with. But it's just it'll pop right off. With the slurp, I'm sure they'll pop off pretty easily. Yeah, it might be nice to figure out what that pink thing is. Okay, so the main target will be the snail.
Okay, and there's another small one right up there.
Oh, perfect.
as
Yeah,
well.
great.
All right, Scott, I don't know how this is going to move once I actually extend, so. Just bear with me.
We're going to slurp these snails and brachiopods.
Looks like the cap got knocked off another one as well. We're gonna close that at some point because there's something. Yeah. Looks jarred.
Yeah. Let's go for 3. Should be good with the mesh.
Tracks, stuff to locate our target. Shows up over here.
Was it just here?
Yeah. First scale first? Yeah.
Yes, please.
You see this guy right here? Kind of right in the middle of the screen? Yeah. I'm just gonna give a wide shot so that you can line up. So, there we go. Yep. that suction on. Got it.
And if you just, Scott, tap these little brachiopods, they might pop off as well.
Okay. Those guys are really fragile, aren't
The shells are, yeah, but there's a stalk that should knock off without too much
Suction
pressure.
still lost.
Great, perfect.
And the pink one?
There's another shell just there,
That little one, right,
Yeah.
in the center of the picture, you see it?
just the top of it.
Kind of buried in the-
Yeah, you
right
could
there.
take
wow.
take
Yeah.
the sediment too. That's why we went for the mesh.
Okay. Yeah.
But you could dig in there and get a bit of the sediment. Actually, that'll be useful
you
too.
want it?
Yeah, no problem. Happy to have the sediment here.
Well, we throw a rock, it's a hard substrate there.
Greg,
Mm.
that's there. That one is there.
Got it.
Got it? Okay.
Nice. That one came.
You want to scrape some of this off too?
Yes, please. And then head over to the pink thing, which we
Okay.
not sure if that's accessible. You could take the brachiopods to try and get at it.
Okay.
We had a request for more brachiopods.
That is bees.
Oh, they're stuck together. They might all come.
Oh no.
Yeah, this might potentially clog the hose.
Yeah, well, they all go.
Ay,
Let's just get that guy.
the pink
Right,
things
just one of the pink thing, right?
We got it. It's a coral.
got it.
So, what else? Anything?
Can we see what that pink thing is there? I know it might be a bit far, but we'll just get an image of it.
Let's see.
Oh, it's a little octocoral.
Okay.
Very tiny.
Um,
There are two polyps.
only two. We'll leave it alone. We could go back near where your slope is.
Just just look odd overview, but okay, it's just the same.
It's the black shell. Yeah.
Alright, what else were you hoping to see?
Can you go up? We can just slope some of the sediment, get a couple more brachiopods from this area. Hopefully they're singles. They'll just come off individually. Oh, there's another shell there too.
Section on scout.
We don't want the coral. Yeah, that's good. Perfect.
And this shell down here?
Yes, please. The other little spiral.
Greg, is it common for these brachiopods to have these sponge features that are attached to their shells?
Yeah, because they're another hard substrate, and so the sponges are happy to settle on them, and the brachiopod might actually stop sediment settling on it.
Do you want more of the?
We could get a couple more brachiopods
Up here.
and sediment. Yeah, thanks, Scott.
It's just one.
Scott, if you could try in here, just, there's a tube coming out between the brachiopods, a little sandy tube. And the animal's pulled in, but if you took the brachiopods, we might get the tube as well. Yeah. Okay.
We took everything.
Okay, hopefully that's all going to go in. And I think we'll call it good. Great. So, Doug, there was a coral in there. Can we just zoom in here again, please? There's a coral Anna wants to look at.
No. Oh, now it's open.
Crab's gotten a piece of brachiopod.
Okay, thanks.
You know what it is?
I'm not sure between, as I said before, Oyabanya or Cariophilia. Looks like Cariophilia.
Paraphilia.
But we need to check inside, and I don't want to collect.
We're not
because it's
checking
only,
it?
No.
Okay.
But we can have a look if we can see others. Maybe.
So, 18.
Yeah, so tiny.
Does this feature keep?
Looks like they have a wall in front of us.
Yeah.
Runs over.
Let's go up a little bit then. Leave the octopus alone. Yeah, that's okay. We've done a bunch of transects, so we've made good progress. Sorry.
Wait, I'm going ahead.
Niskin. Can we just hold here for a sec, please? And let's do a Niskin, please. We only have these ones left.
Yeah.
Okay. Close.
No, the number 4?
I have
Okay, close 5, 6.
5, 5, 5, 6. Okay.
5. Yeah, close 5.
Only 5.
Just one or two?
Oh, you could do them both. Thank you.
There's 5, there's 6.
Okay, let's look at the ledge before we clear it. We might want to try and get the brittle star there.
Greg.
Yeah,
Yes. Look, no.
that's a coral.
Looks like there's no film. Oh, that is so nice.
Okay, nice solitary cup coral there.
Hmm?
Collect?
No, no, it's just one. I don't want. Thanks. But at least we find another one.
Oh, there's a nice worm there. It's not often we see these worms out and about.
Oh.
That's a paddle worm, actually. It's a little sea star. And I think I saw something also over to the left of the brittle star. Oh, it's an isopod. So there's a crustacean related to a roly-poly that you might have in your house. But that kind's often parasitic on fish. Beautiful brittle star, another coral. We might get that if we take the brittle star. We don't-
Okay. If we get it as a opportunistic
bycatch.
sample,
She doesn't mind if it's bycatch.
bycatch.
Okay. What else have we got here? It's a fantastic brittle star. Tube there, yeah. Barnacles on the brachiopods. A little tube worm at the end of the brittle star arm. That's a calcareous tube worm called a serpulet. Little, yeah, baby squat lobster. Yep, yeah, that could be a baby octocoral. A barnacle. Yeah, it's a barnacle with its legs out, not doing anything. Oh, that's a great brachiopod image there. There's another polychaete worm there, poking its head out of the brachiopod shells. It's called Eunice.
Greg,
Yes,
you say that the brachipod has the eggs inside?
yes, you can see
Why?
it.
Yeah, why there are two colors? I saw white.
Male and female.
Ah, okay.
Yeah, they're separate sexes.
Great.
Yeah. So the white little lacy network on the one in the middle is a male, and the yellowish one on the right there is a female.
Okay, thanks.
And they just put the eggs and sperm out in the water, and they fertilize in the water. It's that clear thing, like a little slug. It's not moving. Oh,
It's a juba.
that's a bivalve. That was actually a little skull. Yeah, it was open all the way up.
Coral. Is that coral?
It's a coral.
Coral, black coral.
Black coral?
Yeah, no? Yeah, for the like whip.
It's pretty small.
Yeah, indeed. I wouldn't collect that.
No, we won't.
Too small.
Okay, but I think we could do another slurp here into.
It's rotating.
Oh.
Oh,
yeah.
that was nice. Rotating.
Yes, please. Okay.
They want the isopod.
Yeah, we'll be over here and then probably work our way.
I'm not fully extended, so just let me know when you want. Which chart would you like this in?
Let's go for one, please. Seems like the favorite target is the isopod and the brittle star. So the isopod's there. Oh, it's swimming.
Oh.
It knew.
Oh.
Oh dear.
So, we go back to 3, try to, I can try, yeah. I mean,
Or or you could drop it out. If it's easier.
It was 3, yeah. No.
Yeah. Yeah.
Yeah.
It's only a small. It should be fine then. Isopod got away.
Was lucky.
We could go for the brittle starter, and the
Number.
we'll be back to number 1. Yep. We're on number 1.
Mm-hmm.
And maybe the coral would be good. Cup coral. So that's a hydroid, bushy structure coming out.
Okay.
Well done.
And the coral is there. Yes. He's down. Oh,
Advantage of having 5 arms. Didn't work. And the coral is coming. Yes, coral acquired. Oh, coral's still. Oh, okay. And I think we're good here. What I've noticed is just great. Yeah. And we could just hop up because there's a pretty cool little snail just there.
Are you gonna want that?
Yeah, we
Same
could
jar?
we could put it in the same jar.
Where's the snail?
And there's another one there. I think there's 2 different kinds of snails. Oh, there's the isopod. Haha.
All settled out there. I believe we're going for this.
Yep, beautiful snail. We could get a close-up of it, thanks. Yeah. it's-
oh, nice.
Could be a Calliostoma. Yeah, very nice. Okay. And I think the isopod that swam away is just up there.
Yeah.
Great.
Suction's still on.
And that snail. And there's another snail, I don't know if you can reach. Yes, please. Oh, it's got eyes. Can't quite tell what it is. It's covered with branching arborescent forearms. Okay. Oh yeah, it should fit okay. It's hard to see how big it is.
Oh, yeah.
Nice. Okay. I think we're. Looks like it's coming in, yeah. I didn't see it unless it came through with the cloud. Yeah. Okay, I think we'll stop sloping for a bit.
No, the yellow coral no, but Maritza said that he saw a red coral. I didn't see it. No, the yellow no, because it's similar to the one that we collect. So.
It's not stuck in the tube, or at least as far as we can see. Okay. That's extended all the way out.
Ah, maybe. Ah, maybe that's it. Greg, that thing what? Yeah, can you zoom in? Maybe it's this one?
Yeah, they're a bit busy.
Is this one?
I'll ask.
Ah, I just saw it. Hmm? Ah, por eso yo digo, ¿dónde está el coral? ¿Like this?
Necesito two feet.
Okay.
Looks like Valticina. Yes.
Okay. So we can collect it.
Okay.
Please. Done. Do you wanna record it as the first one?
Can you? Yeah, they would like it. Into a quiver, I guess. I guess you're gonna have to move. Well, you think you can grab it? Great. I think. Could you grab it? Or would suction be easier? We've got plenty of chambers. Okay, let's suction into one of the coarse chambers. It'll have a stalk going in, but it should come out.
It doesn't retract.
No, it won't.
No.
Yeah.
No.
That's a different one.
Yes.
Okay.
Sorry, Scott.
Which one do you want this in?
It could go in. Could go in 8.
8?
Yes, please.
Okay.
Yes.
Thank you.
Yep.
Or the root, you call it.
Yeah, root, stalk.
Stalk. Now let's start. All the way out.
I think it came in.
Just gonna turn that off and see if we can see it drop. And sometimes some really sucked up.
Oh, to the mesh. It was pretty light. I think it would have come through. Yeah, that could be part of it. Just there.
Okay.
Yeah, I think we're good.
Okay.
All right,
Thank
where are
you.
we? Yes, thank you.
Yeah.
Why don't we head towards 20
Yeah.
and then we'll decide if we want to transact. If we see anything along the way, we can stop it. We won't transact. Keep an eye out for things. Looks like it's pretty much everywhere we've been the last hour and a half has just been mud, so it's good to see some life. It's these little ledges and gives enough space for brachiopods to hang on. And then
Hey, corals.
oh, we might get one of these feather stars. Please, yes. So here we have 3 feather stars. They're all perched on a sponge. Could be a glass sponge. We get a nice close-up. There's a brittle star in there, squat lobsters. With the brachiopods, it's a great image. Yeah, it's fantastic. Wonderful. You can see this feather star has 10 arms. It's based on the 5 divisions, similar to what you see in sea stars and brittle stars. A few crinoids have only 5 arms, but most tend to branch once to give 10. And this one you can see one or several of the arms have been bitten off and they've regenerated in the upper part of the frame. Yeah, there's definitely a little regeneration going on. They're pretty crunchy. They've got a calcareous skeleton, but there's still enough food there for a hungry, likely fish to get a bit of a meal. Yeah, there's probably 3 arms got bitten. And they filter feed. They have a channel running down the arm that goes to the mouth, so the mouth is facing us in the middle of that array of 10 arms, but it's protected by all those little cirri, so you can't really see the mouth. And this is mature. It's got those pinnules you can see there with the little feathery appendages coming out are a bit swollen, so they're swollen with eggs or sperm. And it's holding on underneath. You can see the white cirri going out to the sponge to hold it on. Okay, thanks, Doug. That's great. And we might pan a little left and see if there's anything else before we come back to sample. Yeah.
That white octocoral over there?
Next to
Next.
have a close up on the
Yeah,
octocoral.
that.
Yep. That's about the most we can zoom. Yeah. Yeah. Okay. No, if we get the colony, we'll be good. We could pan a little more left and see if there's anything else. That's a dead brachiopod. That's the snail we got before. I think that's a dead brachiopod. Yeah. nicely. Squat lobster, we've seen lots of diversity of squat lobsters the last few days. Quisco Seeps, at least 5 different squat lobsters we saw there. Oh, there's a nice cup coral. Yeah, it's beautiful.
I counted 6 until now. I counted 6.
She's got the eye. Fantastic shrimp there. It's like heterocarpus. Those huge compound eyes. Okay, squat lobster hasn't moved. Let's, got a couple of chambers left. We'll go for number 7. And I think we can select that one of the crinoids. We don't need any more. And we just need the one facing us, Scott.
Okay.
I don't think this is a swimmer, but we'll find out.
We will. Ready for section? Ready.
Yeah, it's,
It's attached.
oh, uh-oh.
Okay, good little now.
I guess that's a drop in a buy box. Yes, please.
Oh.
Yeah, 1A is fine.
One B, 1A.
So you go 1A, or you can go 2.
1A.
These are labeled different. Okay. On there,
Greg. Greg.
So, on there,
1A. 1A.
Not over there, 1A. All the way over there is 1A. Okay.
Not 2B?
Let's go 2A. All right, 2B.
Or not?
Actually, because of on the
Okay. We've been waiting for the audience to notice our BioVox labeling that Lisa and Paola and the pilots did.
Do you need any more infrareds or anything? I'm starting to
Sure. Thank you. Excellent.
Ay. Ay, uy. Push it, push it.
Or I could drop the hose.
Is it
they'll-
no good? Oh, you don't know.
Try not to drop it like that.
It'll fall. Yeah, there we go.
Yes.
Huh. It's good. You subsampled it for us. Thanks. We'll make sure. Yeah.
Are you gonna want suction anything
Yeah,
else?
yeah,
Alright.
we'll still go for the little white coral.
Okay.
Makes it easier.
What are we looking for now?
This little coral there.
Ah, yeah. Still indexed to 7. Ready for suction.
Squat lobster there's got a deformity on its carapace, probably a parasite.
Suction's on.
Oh, I stand corrected. The audience have noticed to be or not to be.
No.
I didn't notice it. So good on you. I think you're doing it. Wonderful.
Nicely done. Salty. Anything else you're gonna wanna suck up?
Yeah, can we have a zoom back in? Just down there, thanks, Dave. Not sure if that's a sea squirt or just a brachydon. Yeah, I think. Oh no, it's a brachydon. Yeah. No, I think we can call that good.
See that coming into the jar or stuck. It's all the way extended.
Let's keep heading to 20, please.
No. Thanks.
You good with the ship
This could be interesting. Wow. Yes, please. Oh, a sea urchin. I like that. Let's go here. Yeah, thanks. This is the first sea urchin we've seen on the dive. Great image. Just getting what? Exposure right. Yeah, that's great. So if you remember before, I said this is the closest relative to sea cucumbers. I'm not sure of the name of this, but we'll figure it out. Quiver? Please.
It's like
637. Yeah. How often do you change out the little silicon fingers? The day before one? Yeah.
It's fortunate for us that they're just some degree print out here, so we can take them a little.
Right.
Yeah.
Look at that. Masterly. So you can see the little tube feet of the sea urchin that it uses to walk with and protect itself, as well as the spines. So we'll take this for identification, genetic sequencing, and it will go to the National Museum in Santiago at the end of this cruise.
back down and get the handle out of the way.
That's the only thing you can do, because you can't see.
Yeah, we have to put it down there. Oh, the camera views couldn't show Scott how to put the urchin in the container, so the lid was blocking it.
What's the matter?
The handle is up.
Oh,
Just right over it.
Oh, you can see it with the T4 camera. Sorry, I'm watching the T4 camera.
Right over the container now. And it's in.
Yes.
Well done, thank you. Well, I'm going to hand over to our chief scientist, Elohio Soto, now for the next few hours as we continue our transect up this section of the San Antonio Canyon. Hopefully talk to you tomorrow. Thanks, guys.
Where would you like to go, sir? We were heading towards, we had 120, but you tell me where you want me to go.
Let me check.
So we were on our way to this waypoint. But wherever you'd like to go, just so we came from up here, right? And they basically just there was a bunch of flat ground, so we found this vertical spot. We were just following this vertical spot. That's what we were doing.
So we can move to the waypoint.
Okay, no problem. We'll head that way.
Thank you.
Sure.
Thank you. Hello, can I speak now? Hola, buenas tardes. Les habla Eulogio Soto, jefe científico de la expedición. Nos encontramos a bordo del barco oceanográfico Falcor 2 del Schmidt Ocean Institute de Estados Unidos. En estos momentos estamos haciendo una prospección biológica y estamos estudiando el cañón de San Antonio. También se le podría llamar el cañón del río Maipo en la zona central de Chile. Nos encontramos a 798 metros de profundidad y hemos estado siguiendo un transecto en zigzag con el objetivo de conocer y describir las comunidades de invertebrados y peces que habitan el contorno de una ladera que escogimos en este cañón. Nos hemos encontrado con comunidades muy interesantes de invertebrados como braquiópodos, corales, actinias, crustáceos y otros organismos. A bordo de la embarcación se encuentra un grupo diverso de investigadores de distintas disciplinas, algunos de ellos geólogos, geofísicos, pero también biólogos marinos y oceanógrafos. La expedición está liderada por la Universidad del Paraíso, pero también está liderada por la Universidad de Boston, la Universidad de Aveiro, la Universidad del Estado de Oregon, y también participan otras instituciones de Chile y del extranjero. Ha sido muy interesante el poder conocer este cañón. Nunca antes en Chile se habían podido realizar estas filmaciones en directo y con esta calidad, con esta resolución, lo cual nos permite no solamente recolectar organismos, sino también poder ver conductas, adaptaciones, formas de vida, tamaños in situ, es decir, en el mismo lugar. Una de las formas de investigar un cañón submarino puede ser siguiendo el contorno o una ladera y seguir un recorrido que permita ir observando las diferentes comunidades, hábitats, formas de vida. En este caso hemos elegido el contorno sur. Comenzamos a los 1100 metros y hemos estado subiendo en una pared con cierto grado de inclinación para poder ir tomando muestras también de los sedimentos y caracterizar la química sedimentaria. Pero principalmente nos ha interesado hoy día ir conociendo y describiendo la presencia, la distribución de los organismos que habitan en torno a estos ecosistemas de cañones submarinos, en este caso el cañón de San Antonio. saludamos a todos los que nos están viendo desde la plataforma streaming del Schmidt Ocean Institute o que nos están escuchando en directo a través de esta transmisión. Luego de terminar esta inmersión en este mismo punto, realizaremos estudios con CTD, con instrumentos que permitan determinar la conductividad, la temperatura, la salinidad de la columna de agua. Y también durante la noche se realizarán lances para estudiar las comunidades del zooplancton en el cañón.
You got two open. Which one do you want to do or something?
Maybe 5?
5? It's got some. Is that not true?
5 is
Okay,
up.
you have 5 open. Okay.
5 is available.
Yeah, we both have something in 5. Is that the case?
We don't have nothing in 5. Wait, just a second.
Okay. Wait to see. First shift.
They have a secret camera in the first shift.
5 is available.
Yeah, they have a secret camera in the first shift. Yeah, mine is empty too.
2?
No, this is empty.
Okay, 5.
5. Five words going. Ready? Ready? 1, 2, 3. So, I thought you were a little higher than that. Yeah, me too. Fight. There goes.
Wow. Good.
Okay, so next time I come all the way out.
Octopus.
It is all the way out. It is on there.
Thanks. Thank you.
You can, put it, the way I say it, I think that's the last chart, I think is.
Thank you.
And then, deadline did jar 5, right? Jar 5, yes.
Bueno, acabamos de recolectar a través de la bomba de succión o de la succionadora un pulpo en este hábitat donde también podemos encontrar corales, bandas de braquiópodos y también tubos de poliquetos y algunos cangrejos. Todo el material recolectado será posteriormente identificado y formará parte del patrimonio natural que será depositado en el Museo de Historia Natural de Santiago. Cada grupo es revisado e identificado por expertos, en este caso, llevando.
If you want to go to 20, I have to go back down, can't you? If you want to go to 21, I go up, can't you? The wall.
We need to
So, this way is down, this way is up.
20.
You wanna go to 20?
Yeah, 20.
Okay, we're gonna go downhill. Alright.
I go down?
Down, there we go.
Okay.
You good with the ship? Where is it?
with my camera, please? When it's
So,
possible?
so
Thank you.
21.
21, okay, go on uphill.
21.
Yeah, okay, up. Yep, power roll. Yep. Alright, Scott, we're going up, so you. Okay, that's all, okay. And power roll is cloud. Thank you.
Hello, really? We got updated waypoint for you? Alright, cheers.
Heading towards 21. Up slope. Yes, he's.
Ah, sea star.
Seeps. I'll be there.
Thank you.
All good?
Yeah, good. You just kind of lost connection, and I'm.
Some coral quivers. Bueno, retomó la narración después de acá con los pilotos del Sebastián, más que nada. A veces hay que conversar acerca del recorrido, del track que uno va siguiendo. Entonces nos consultaban cuál era el próximo punto y en base a lo que hemos planificado estamos siguiendo un transecto en zigzag. Entonces ahora nos dirigimos al punto 21. Ahora podemos observar una estrella junto a una pareja de corales y vamos a recolectar esta estrella. Como les decía, cada grupo faunístico es identificado por expertos, lo cual toma un proceso en algunas ocasiones bastante largo, con el objetivo de identificar correctamente cuál es la familia o el género o la especie. Y en algunos casos, si llegara a ser una especie nueva, bueno, los ejemplares deben ser descritos y posteriormente depositados en el Museo de Historia Natural de Santiago. Todos estos esfuerzos también son colaboraciones con distintos investigadores que participan de la expedición, que nos ayudan a realizar estas investigaciones. Follow to the toward. Les contamos también que luego de terminar este estudio del cañón de San Antonio, el cual se extenderá hasta el día de mañana, seguiremos hacia el sur, hacia las filtraciones de metano registradas en la zona de Concepción. También procederemos a estudiar el cañón del Biobío. Y posteriormente nos dirigiremos a la isla Mocha para prospectar y ojalá poder encontrar alguna filtración de metano en torno a la zona contigua a lo que es la isla Mocha. Sabemos que existen antecedentes de emanaciones de gas en la zona costera, pero se realizarán los mapeos necesarios cercanos a la isla para poder encontrar un ecosistema en una zona mucho más profunda. Una vez terminados los estudios en torno a la isla Mocha, el crucero se dirige hasta Puerto Montt, donde recalará el día 4 de noviembre, dando fin a esta primera etapa de este proyecto que tiene por objetivo estudiar los cañones, las chimeneas hidrotermales y las filtraciones de metano del margen continental de Chile. Posterior a la recalada en Puerto Montt, la segunda etapa continuará hacia el sur, dando prioridad al estudio de cañones en el sector de la Patagonia, pero también dándole importancia al estudio de un ecosistema en donde se juntan tres placas tectónicas, que es llamada la zona de la Triple Unión, en donde esperamos poder encontrar una chimenea hidrotermal activa. Estos ecosistemas quimiosintéticos muy interesantes, muy extraños, en donde en algunos casos es posible observar estas emanaciones de gases de alta temperatura y junto a ellos, bueno, una vida muy distinta a la que generalmente uno suele ver en el mar profundo. El crucero continuará mapeando y investigando cañones al sur de la Patagonia para finalmente terminar en Punta Arenas la primera semana de diciembre. Es interesante comentar cómo se distribuyen en bandas horizontales, en este caso muchos de los organismos que podemos ver en estas laderas o contornos del cañón. Es el caso de los braquiópodos, así como también de algunos corales, forman verdaderas bandas de distribución en el sentido horizontal que se van disponiendo a lo largo del cañón.
Hey, on the bridge, Eric, did we go?
Out Okay, thank you.
Most.
Yeah, two different kinds.
Oh, yeah. Would
Yeah.
make it. Can you take notice that you structure from the native just like this?
So if you look here, it looks like it goes that way all the way back. If we break that off, it's just gonna fall on top of it, I think. Scott, you got an opinion on that? Look at the structure. You want that worm, but it goes up underneath. It doesn't go like, you have to grab that whole cluster. I don't know what happened with that. Might just crush it.
Try to fingertip grab the worm.
No, you just want the whole, like, to go back, like, grab the whole structure, see, the whole thing. It looks like it goes, it weaves back underneath. See what I mean?
Right.
Up under there. But he's thinking if you grab this, it will come off, but I don't know. I think
Yeah.
it'll just pull off and the rest will fall. We could try. We could
Yeah,
try.
yeah.
Bio box though, if we get it? Yeah.
Yeah,
Okay.
bio.
Okay.
Go with $4.
Want to try with just $4?
Grab the whole thing.
Maybe.
Starboard.
Yeah, I'd see where your closest. Your closest to starboard, it's over there.
Okay, starboard.
I can get you closer if need be. It's just like, clams and top clams. Yeah, it's already sunk itself in there. Yes, yeah, just try and get to the right of it just a touch, maybe break the whole cluster off. Yeah, like right in there. Yeah, you got it now though. But it's all falling apart. So there's no telling, there's no telling if it's in there.
Could be fine.
I don't know, maybe flip it around. Is it hanging off the other side?
Looks like it is. Just go with the box.
we can keep it anyway.
Want that? Oh, I see what it is, just something.
Perhaps we can find some animals in.
You want what's in the ball, or no?
Oh, I think we can keep.
Okay, no problem.
Yeah.
Go ahead and work it out.
I guess the retrospect maybe is scooped up, I guess. Hold it in order for.
Just scoot it down a little here. Live box 2B.
Yes. Yep. Biobox BB.
Yeah.
We follow to the 22.
Covering 7 to
7, yeah, I believe so.
Okay, stop. Can you make it this? You can collect this one.
Okay. But that's,
Maybe coral colors 7
Might be a little bit big for a
7 or 8.
If we open it, it doesn't fit, then it was an option.
Elogio.
We have
20. Don't remember putting anything in here. We put something in there, but that one, maybe they did earlier.
snail. There is a snail,
Oh, okay.
starfish,
Okay.
sea urchin. Una de las investigadoras que se encuentra con nosotros es la bióloga marina Fernanda Orrego de la Universidad del Paraíso, quien saludará y contará más detalles de su trabajo en la expedición.
Bueno, yo estoy a cargo del muestreo de zooplancton, el cual es realizado mediante la retorno. Las muestras son fijadas, guardadas y después son identificadas y su posterior análisis se hace en el laboratorio de ictioplancton y interacciones biofísicas de la Universidad del Paraíso, la Facultad de Ciencia. Actualmente yo trabajo en este laboratorio principalmente con morfometría geométrica.